Good morning, everyone. We're going to give this about another minute or two as the number of attendees rises here this morning. Okay, we'll give it about another minute here. We're approaching 150 attendees. Good morning. Let the coffee hit those neurons, and we'll get started in under a minute here. Okay, let's fire this up. Good morning, everybody, or good afternoon, good evening, wherever you are in the world. Hello, and welcome to Biotech by the Lake, 2021 edition. My name is Neil Kelleher, and I'm your host today. I'm also the acting director of the Chemistry of Life Processes Institute here at Northwestern, and the director of Northwestern Proteomics. Though we missed the opportunity to meet and commiserate all in person, as we did before the pandemic, we're incredibly pleased to welcome so many of you, from investors to industry leaders and innovators and investigators from across the US and beyond. First, I'd really like to thank Oppenheimer for their support and partnership this year, and especially Jay Olson, a research analyst at Oppenheimer, who will be an active participant and has been instrumental in developing today's program. Jay will lead the neuroscience, biotech financing, and investing panel discussion later in the program. We're also really grateful for the team at BioCentury and Lauren Mertz, her senior editor and head of clinical translation and development at BioCentury, who will moderate today's panel on hot topics in neurological disorder therapies and technologies. I'd also like to thank the CLP Institute's Extraordinary Executive Advisory Board, who were key in launching this program 3 years ago. I'd especially like to recognize Sujul Shah, President and CEO of Theamyloid Therapeutics, who originally inspired us to reach out to Oppenheimer and tee this up. So today you'll meet another valued member of our board here at the Chemistry of Life Processes Institute at Northwestern. And that's Mi-Young Shin from Merck, who will participate in the hot topics panel discussion led by Lauren. Before I turn it over to the speakers in just a few minutes, I'd like to provide an overview of our institute here at Northwestern, Chemistry of Life Processes. It was chartered by Northwestern in 2004, and we were planning a campus research building those many years ago. And it was designed to drive collaboration among chemists, engineers, and of course, life scientists. A few years later, we launched the Richard and Barbara Silverman Hall for Molecular Therapeutics and Diagnostics. And this was completed in 2009, thanks to a generous donation of Rick Silverman and his wife, Barbara. And he's a founding member of the Institute and, a reasonably talented chemist. Rick has donated a portion of his royalties from the sales of Lyrica, a blockbuster drug that many of you know was first approved for the treatment of fibromyalgia and diabetic neuropathy. You'll hear from Rick today and his newest innovations, because lightning does strike more than once here by the lake. And he's active in treatments for ALS and Alzheimer's. So you'll hear that a little bit later. This is Silverman Hall, as you can see, and it's written on the building, Diagnostics and Therapeutics. So 16 years later, we've established an extended ecosystem for drug discovery and development at Northwestern with 65 faculty across 20 different departments at Northwestern. And we have appointments ranging from Northwestern Medicine, McCormick School of Engineering, Weinberg College of Arts and Sciences. We work transdisciplinary across boundaries, and in team science to open up new avenues of discovery and treatment of disease and to promote health and wellness. Our goal is to make tangible differences in people's lives, and we're doing that. And clinical translation is a key part of that. And we lower the barriers to discovery and catalyze the early stage research across the chasm that many of us know about in this business. And with our colleagues at Feinberg School of Medicine, we bring innovations from the lab to society. We have various award mechanisms that do that to empower young and older teams of scientists here at Northwestern. And we've got about a 20-to-1 return on investment back to our institution. Moving on, we have a lot of infrastructure and a solid foundation from which we build. We have a core of research administrators and staff. That's the backbone of the Institute. And we've secured more than $60 million in external funding. But what really distinguishes CLP from many of our peer institutes across the country and world is our technology platforms and resources we built up over the last decade. And we're home to several university centers and cores for drug discovery and development of diagnostics that are open to faculty across the university and indeed far outside the walls of our institution. We've made significant investments to provide comprehensive tool set that enables our faculty, postdocs, graduates, and undergraduates to pursue science with low barriers. We employ more than 60 researchers and technical staff to accomplish this. And we've really put a strong emphasis on making inroads to understanding neurological disorders and other diseases, and you'll see that momentarily. And as a last point here, we've spun out 27 new companies to commercialize discoveries and attracted more than $2 billion in external investment. And the intellectual property behind a very promising Alzheimer's therapy, currently in development by Acumen here in the top center, was developed by one of today's speakers, Bill Klein, and we're just getting started. So you'll hear about that more momentarily. And, particularly with today's and even this month's press and lots of discussion, we have a really timely program today. And all the dynamics of the industry about neurodegeneration, neurodegenerative disorders, the recent FDA approval of aducanumab, and many pondering a re-entry into this space as we really push down on the accelerator to get at the causality of neurodegenerative diseases. So it's an exciting time, and many of these developments are in the first part of our program. Okay, so without further ado, let me just take care of a couple housekeeping notes. After each presentation and panel discussion, we'll take a few questions from the audience as time allows. And to ask a question, please use the Q&A icon at the bottom of your screen to submit questions, and we'll field those and present those. We'll select as many questions as we can and share them with the speakers for their response. And if you'd like to post about your experience today on Twitter, we're using the hashtag Biotech by the Lake. So welcome, and indeed, let's transition to our first speaker. We've got 4 great speakers in the first part of our program, and the first of those is depicted in the upper right. That's Hande Osdenler. There's Bill Klein in the lower left, and we'll get started. So I'd like to introduce P. Hande Osdenler. She's an associate professor of neurology here in the Feinberg School of Medicine at Northwestern, and her title is Improving Success Rate of Clinical Trials by Proteomics. And please, Hande, take it away.
Thank you so much. And I'm trying to see if I can, yes, here I am. Okay, wonderful. Thank you so much for the introduction. And I'm very happy to be here. And as you said, I think this is an important time to form the ties and the connections with the scientists and the industry. And I will be talking a little bit about upper motor neurons and how we can improve the success rate of clinical trials by using proteomics. So first, if I make an outline, I will briefly mention the definition of the problem or the problems, And what are the unique strengths we have at CLP and in our lab? What is the path and strategy to move forward? And what are the opportunities that we have? And as you mentioned, diseases of the brain are a major unmet need. We don't have any effective cures for any of the neurodegenerative diseases. Even though the developments are emerging very fast, we still don't have a feasible and effective treatment to any of the neurological diseases, and the numbers are increasing. And of course, patient population is increasing, no cures, patient population is very heterogeneous, clinical trials keep failing, and that's a big problem. And the burden on society, on economy is increasing. We expect to have even larger number of Alzheimer's disease patients, from 5 million to 14 million is expected. And the same is for Parkinson's, the same is true for ALS, especially we're expecting a 96% increase by 2040. So it is because patients are diagnosed more effectively, patients live longer, and now we have a huge unmet medical need. And of course, if you think about the rare diseases, they're not really rare if you put all the rare diseases together. It is a large patient population that we have to deal with, and the numbers keep increasing. So our lab focuses on upper motor neurons, and these neurons are exceptionally important because they initiate and modulate movement. And their degeneration is seen in ALS, HSP, PLS, and the long-term paralysis that is observed in spinal cord injury patients is also in part due to the degeneration of the upper motor neurons. So if we find a way to help them or to cure them, we would touch the lives of thousands, millions of patients in the United States and around the world. So the upper motor neuron diseases, for example, the ALS patients, it's 50,000 new patients every year, 86 per 100,000 children in the US born with HSP. So these are not really small numbers. And imagine, it's not just one person, this affects the whole family. So for example, the PLS, 2,000 new patients, and in spinal cord injury, there are about 20,000 new injuries every year. And again, no effective treatment strategies, even though there are a couple FDA approved drugs, but they extend the lifespan for a short period of time. And most of the clinical trials actually fail on phase 3. So the mechanism are very heterogeneous. There are many reasons why patients develop the disease. And now we're beginning to realize that one drug is not going to cure all. And the FDA actually moved on to accept the expanded access, off-label use, right to try, so that patients can actually have access to other compounds who have passed phase 1 and phase 2 and failed phase 3. So we are moving towards personalized medicine approaches. So this is a new time for drug discovery. It's not going to be one drug for all patients. We're going to develop a medicine around the needs of the patients. And that's why we need to understand the rare disease patients, which are making a huge patient population. So we need to understand more personalized approaches. And in the conventional treatment, normally what happens today is they take a huge heterogeneous population and they give one drug, but of course some will show an effect, some no effect, and some adverse effect. But then FDA says, well, this is not clinically significant, and the trial fails. But if we were to move from this picture to this picture and delineate patients based on the mechanisms that are responsible for their degeneration, and then very cleverly give them the drugs that actually would help the mechanism that is responsible for their degeneration, then we would have very successful clinical trials. So then the bottom line is going from this picture to this picture. If we can manage to do this, then we will have multiple successful clinical trials, and we will be able to lay the foundation for personalized medicine approaches. And I think that's what the time is getting ready for. And what is the limiting factor to do this? Understanding the mechanism. And how can we understand the mechanism in each patient? So we propose that the proteomics and biomarkers are exceptionally important, and they will allow us to move from this picture to this picture. And I'm going to tell you briefly how we can do this. And once we do that, the layout is going to change so much that we will develop mechanism-based treatment strategies, so that the same mechanism may actually be shared by Parkinson's, Alzheimer's, ALS, other rare disease patients. So then rather than curing a disease, we will cure the mechanism. And then by curing the mechanism, the drugs that we discover are going to have a broad perspective or broad implications for many of the diseases. So we won't be looking for one drug for one disease. We will be looking for one drug or multiple drugs for different mechanisms, and then we will match make these patients based on the mechanism that goes wrong in them, and then sky is the limit. So then there won't really be any rare disease annotation, because for each disease we will know what goes wrong with them, and for everything that goes wrong we will actually have a solution. And I think that's what the 21st century is asking us to do. This is how we should move forward. And what is really good is that CLP and Northwestern have a unique strength to overcome this limitation. So we have an immense potential to have successful clinical trials, to treat more patients, to develop effective treatment strategies, also for rare diseases. And millions of patients are left behind because they are rare disease patients. But now it could be that there are no patients left behind and everyone is included into the treatment strategies. And we think proteomics and biomarkers are the way to go. Of course, there are many different biomarkers. They could be diagnostic, predictive, pharmacodynamic, and you can isolate the biofluids from serum, plasma, CSF, urine, and even the diseased neurons. So then, of course, there are proteins that would go up and down, and they would tell you if the patient has the disease or not, or some proteins may be lacking a distinct portion of the protein. So it's a splice variation or post-translational modification differences. And these proteins are very important to reveal the biology. So how do we go about this? So first, we made the upper motor neurons visual. I'm going to go very fast because these are all published results. First, we made the upper motor neurons visual. We made the first reporter line. We crossed them to many different disease models, and each of these models represent a mechanism, okay? So then now we have diseased upper motor neurons that become, that are diseased due to a known mechanism. So once we do that, then we can actually FACS purify them, fluorescence activated cell sorting, at different times of the disease, and then we look at the proteomic profile. And by doing so, we can actually investigate both top-down and bottom-up approaches. And we've been working with Dr. Kelleher and Young. So when we do bottom-up, it's basically digesting and then reading the protein. But top-down is more important because it gives you the proteoform structure, because that includes the splice variations, post-translational modifications. A protein is not only a protein. A protein has dresses. It dresses up. It has all these additions, and those additions are critically important for their function. So it may not be the lack of protein. It may be that the protein has a different domain or missing a domain, and that doing top-down would actually reveal that, and that's very important and has not been done, but I think that's what the future calls for. Of course, now we have different models, and we also collect blood. We do the same thing, proteomics, but what is really nice being in a medical school is that we also have access to blood samples collected from ALS patients who have SOD1 mutation, who have TDP pathology, alsin, profilin, you name it, right? And then these are, some of them are longitudinal samples. So then you may imagine that there is really not much common between mouse and human, but actually important proteins are shared. There are some common ones. So those are extremely important for the biology of the disease. And of course, we perform proteomics. They tell us the canonical pathways that are important. And talking about the canonical pathways, there's another way that we can look in the protein-protein interactions and how they play a role in canonical pathways, because these are the mutations detected in ALS patients. So we have revealed that lipid homeostasis is important, protein homeostasis is important, and DNA damage repair and cytoskeletal dynamics. So these are all published work, but by showing this, I also want to tell you that the proteins that are present in human and in human DNA, mutations, their interaction, and combining it with long-term proteomics is extremely important. We have the information. And looking at the longitudinal samples, there are some proteins that go up over time. So this would be very important for pharmacokinetic analysis. So it will also help us understand the disease mechanism. Let's say if you have that protein, it's increasing, or most likely you actually have this mechanism. And most likely this is how much your upper motor neurons are degenerating over time. So the knowledge was lacking, but now we actually are beginning to establish that knowledge, and that lays the foundation. And we have established the first upper motor neuron research lab. We have extensive knowledge on upper motor neuron biology, largest collection of transgenic upper motor neuron disease models, one of the best proteomic centers in the US, world leader expertise, especially for top-down proteomics, largest serum and plasma collection from upper motor neuron disease patients in the world, and we have access to postmortem human brain samples for confirmatory analysis later on. Therefore, I think our unique strengths creates a unique opportunity, and that's what we would like to identify in the future. We would like to identify biomarkers that would tell whether the patients have the disease, and what is the underlying mechanism responsible for the disease formation, and what is the timing and extent of upper motor neuron loss. This is extremely important for clinical trials to see if the treatment is working, and I think we could reveal that information. So coming back to this picture, we are stuck here, and our goal is to go from this picture to that. And I think we have the scientific expertise and the tools and reagents ready for this. And for today's meeting, our goal is to establish strategic partnerships with you, so that we together invest in our collaborative efforts and help develop the first biomarkers for upper motor neuron diseases. And especially for upper motor neurons, there is really no cure. This is an untouched territory. And our unique strengths in this field makes us it gives us a opportunity to succeed in that field. And I think we can improve the success rate of clinical trials, and we will help treat rare ALS disease patients. So with this, I would like to thank you, and I will be looking forward for your questions. Thank you so much.
Thank you, Hande. That was really well done and fast. That was great. So we have time for questions, and I'll take the MC's prerogative and ask you about, the upper motor neuron type diseases. What diseases are tied to these subtype of neurons, and what is known about their mechanistic contribution over time and in those different diseases?
Yes. So first of all, upper motor neurons have been mostly ignored in the field, because there was this notion that there is dieback in ALS, that first it's the neuromuscular junction, then the spinal cord, then the corticospinal tract, and then the upper motor neurons. So nobody actually thought that upper motor neurons would be clinically relevant or clinically important, and they didn't really think that they were targets for therapeutic intervention, because they were going to die anyways, right? But now we realize that actually the disease starts in the brain. The cortical hyperexcitation occurs 6 months before disease onset. So the spine loss is one of the first things that happen in the upper motor neurons. And apical dendrite degeneration is one of the first things that happen. Therefore, we now know that if we cure upper motor neurons, we are going to cure the motor neuron circuitry, because the movement starts in the brain. And that gives us a unique strength as well, because if we focus our attention to upper motor neurons, this will have implications in ALS, in HSP, in PLS, even in spinal cord injury patients. So the upper motor neurons are critically important for any disease that require voluntary movement and that require motor function. So I would imagine that the next 10 years would be the upper motor neuron year.
Yeah, and there's a follow-on question from Israeli. Have you done a dry run for the proteomics, FDA-approved drug mutation match in an animal model?
We have, and it's not complete, so I don't know the results yet, but yes, we are working on that. And I would like to talk to you more about this.
Yep, so I think it's teed up and moving along. There's another one from an anonymous attendee. You fed all the neurodegenerative diseases into mitochondrial dysfunction. Are you exploring more upstream targets in each disease state, or focusing more intently on how each translate to mitochondrial dysfunction?
Yeah, so in my figure, I just use mitochondrial dysfunction as an example. So it's not really, maybe I should have explained. Mitochondrial dysfunction was just one example. There are multiple common causes. For example, one of them is the cytoarchitectural defects. There are problems with axon transport, actin tubulin defects that is all observed in many neurodegenerating neurons. The other one is the lipid homeostasis. So then, for example, especially in the case of Alzheimer's, we see that the neurons cannot maintain their lipid homeostasis. That also links to the post-translational modification of proteins because there are problems in the Golgi. So mitochondrial dysfunction was just one example because it's also shared by Parkinson's disease and some of the Alzheimer's disease as well. So I think we need to look for the common causes of neurodegeneration. And then focusing on the common cause, then we can branch out to whichever neuron dies due to that common cause. And there are, I'm sure, engineers in that call. Engineers look for the the root cause analysis, right? They do the root cause analysis, and they don't really, and doctors really don't want to treat just the problem. We want to go to the deep core of the problem. So the if you do the root cause analysis, you will see that all these causes are actually common, but they are different combinations in different patients. So if we do treat the root cause, I think that would be the solution for neurodegenerative diseases.
Okay, and we'll go with one more about Nicholas wants to know if you used other cores for your research. Like, what other infrastructure do you funnel into this to try to address along the vision that you laid out?
Yes, so of course, at CLP we have the best score, so we didn't really have to go out for others, but we also use metabolomic core facility because in addition to RNA-seq proteomics, we also do metabolomic analysis. Now we're beginning to do lipidomics. So that's also very interesting because the lipids change over time, especially for a promoter neuron. So we are trying to cover most of the omics based on the pure neurons and serum and plasma over time, different mechanisms. And I think that's going to give us the full picture.
Okay, great. Hande, that was awesome. Whet the appetite here and some neuron lubrication as we get the program going. Really, thank you so much. We're going to turn it over to our next speaker. And so we'll advance the slides. Yeah, thanks, Hande. And that's Bill Klein, who's probably known to many of you. And he's professor of neurobiology in the Weinberg College of Arts and Sciences. And with no further ado, Bill, please take it away.
Thanks so very much, Neil. It's really terrific to be here. I'm going to be talking with you about the next generation of Alzheimer's immunotherapy this morning, and what the focus is on targeting the toxins that actually, in fact, trigger brain cell damage that leads to Alzheimer's disease. As Neil alluded to a minute ago, there's big news in our field, and basically it's hot off the press. A week ago, the New York Times already had its front page headline on Biogen's new drug, where the FDA has given tentative approval, actual approval, to aducanumab, which they're calling Aduhelm. And this is the first new approval in 20 years in the Alzheimer's field. And it's also significant because it has a therapeutic that's aimed at the toxins that cause Alzheimer's disease. And as you see down at the bottom of this headline, there is residual uncertainty. As a matter of fact, there's been huge disclaimers from the advisory board about this, but in fact, today's Wall Street Journal had pushback on this. So I think what's happening is that we're not quite there yet in getting to the target of the toxin, but we're getting close. And in fact, there's been a hunt for toxins since Alzheimer first examined the brain of his patient, Augusta D, back in 1906. And you're all familiar with plaques and tangles in the Alzheimer's brain. And Alzheimer was the first to see both of those in 1906. This is a more modern picture of it. The amyloid plaques, which you may know are extracellular deposits of abnormal aggregated proteins. That's shown in the center with the A here. These little black areas, those are neurons that are filled with neurofibrillary tangles associated with the protein tau. The amyloid is instrumental to the formation of what's been the most powerful hypothesis in the field for decades, the amyloid cascade hypothesis, where a membrane protein called amyloid precursor protein gives rise through proteolysis to a small peptide, 42 amino acids long, called beta amyloid, or A beta. And that, in fact, as it reaches higher concentrations in the brain, fortunately, even though we're all making it right now, our brains are getting pretty good at clearing it out. But if it doesn't clear it out, it self-associates and forms fibrils, which precipitate and form amyloid plaques. And the amyloid cascade hypothesis has said that neuron damage leading to Alzheimer's disease is due to these amyloid plaques. And of course, what one would want to do would be to target those amyloid plaques based on this theory. And in fact, there have been vaccines, small organic molecules, and therapeutic monoclonals like Biogen's aducanumab. Unfortunately, these have been tremendously disappointing, and it's been a real brake on the field's development, the amount of money that's been invested and not lost. But we're moving forward in a number of directions. First of all, I'd like to say that one of the reasons for these failures, among others, is that focusing on amyloid is the wrong target. The right target, I believe, is the amyloid beta oligomer. What you're looking at here is at atomic force microscope images of synthetic amyloid on the left and synthetic oligomers made on the right. They're both actually made from the same starting material, an A beta peptide synthesized and then put into a test tube and allowed to incubate for 24 hours. Under one condition, you end up with amyloid fibrils. Under the other condition, you end up with soluble A beta oligomers. And this observation was made by my colleagues Grant Kraft and Finch and I, and interested in the properties of these, as I'll tell you about, which led us to start Acumen Pharmaceuticals over 2 decades ago. We were interested not just in the structural biology of A beta oligomerization and fibril formation, but the fact that we can attribute toxicity to these molecules. In our paper that we published in 1998 in PNAS, we reported that fibril-free oligomers impaired synaptic memory in the form of LTP and caused a selective nerve cell death, basically the underpinnings of the disease mechanism in Alzheimer's disease. So we proposed the A beta oligomer cascade, memory dysfunction early on and progressive dementia during end stage could be caused by A beta oligomers. A terrific example of illustrating the toxic potency of these A beta oligomers is an experiment where we inject A beta oligomers, fibril-free, into the brains of mice and take a look at their ability to recall what's new and what's old. This is a novel recognition task. And on the left, the mice are injected with a vehicle, exposed to a couple of objects. A day later, one of those objects is moved. And mice, being curious about new things, spend most of their time looking at the object in a new place, unless they've been injected with A beta oligomers. These animals can't tell the difference between the old object and the new object. And you can actually switch the object as well. And instead of just the position, you can change one of the objects to a new shape. Animals injected with vehicle are very good at recognizing what's new from what's old. Animals that have been injected with oligomers can't tell the difference. And in fact, this same type of behavior shows up in transgenic animals, such as the 5X FAD mouse, which was developed by Bob Vassar's lab in Feinberg. This animal develops oligomers spontaneously after a couple of months, and then at the same time loses the capacity for this memory task. Since we published on A beta oligomers, there have been over 4,000 papers in this field because of the fact that this molecule can actually be upstream from essentially all of the downstream mechanisms in Alzheimer's diseased brain. Tau phosphorylation, for example, or selective neuron death, or synapse degeneration. These can all be triggered experimentally by A beta oligomers, either in cell culture, in mouse models, transgenic models, or in fact in a recently developed non-human primate model. Now, I underscored the word experimental, and the question is, does that experimental approach translate into human brain? Are these oligomers, in fact, disease relevant? And we established that that indeed is the case. We're looking at human neuropathology here. 3 specimens, one from an aged human brain without dementia, one from a aged human brain with intermediate levels of dementia, one from an aged human brain with advanced dementia. And these are being stained with an antibody that is specific for oligomers. So the immunohistochemistry, immunofluorescence microscopy here uses these oligomer-specific antibodies, which in fact were first developed at Northwestern 15 years ago. We also see a soluble extract assay showing the same thing: non-cognitive impairment, no levels of oligomers, Alzheimer's disease with our antibodies, high levels of oligomers. So developing these oligomer-specific antibodies for research purposes, in fact, got us off in the right direction for therapeutic purposes, as we'll see in just a second. So we also can do biochemistry, immunochemistry with these oligomer-specific antibodies. Up at the top, you're seeing a night and day phenomenon where Alzheimer's brain extracts are spotted on a piece of filter versus control brain extracts on the right, and stained with our oligomer-specific antibody. And you see the disease-dependent buildup of the oligomers. And if we look at the structure of those molecules, which is important to confirm the validity of our experiments, if we do 2-dimensional Western blots, separating first by isoelectric point, then by mass, and then do a Western blot, we see that our oligomer-specific antibody in an Alzheimer's frontal cortex extract stains roughly 12 mers at an isoelectric point of 5.6, the same as we see in synthetic A beta oligomers, which we called ADDLs once upon a time, and essentially absent from the controls. The point being, brain-derived and synthetic oligomers structurally are homologous. We have found these not just in brain extracts per se, but also in the cerebral spinal fluid of Alzheimer's patients. And this is an ultra-sensitive nanotechnology assay that Chad Merkin developed, and we collaborated with for assaying A beta oligomers. This is part of the Nanotechnology Center as part of CLIP at Northwestern. Tremendous nanotechnology capabilities. And what we found here is that we're able to detect a 30-fold difference in the spinal fluid between Alzheimer's diagnosed patients and control populations. It is a semi-log plot to indicate this is the difference. This establishes another proof of concept for the presence of these oligomers in a disease-dependent state. Also shows that they are extracellular. And in fact, also shows that this is a potential biomarker. So extracellular oligomers, what might be the mechanism? What I'm showing here is a cultured neuron, hippocampal neuron, that has been exposed to our test material. In this case, the synthetic A-beta oligomers. The unbound material, if there is any, is washed away, and then we apply our antibody probe for oligomers. And these little gold spots, these hot spots, are in fact the binding of the oligomers to synaptic spines. If you looked at it very closely, these are at synaptic spines, and this is what gets the mechanism going. An important experiment that I especially like was to see whether this carries over to human brain extracts. And in fact, if we take healthy brain extract and do this experiment, we don't see any binding to these dendrite synapses. This is a little bit of glow from the secondary antibody in the cell bodies. However, if we take an Alzheimer's brain extract, and not even purified, but a high-speed spin, what we find is this same dendritic binding at synapses as we see in the synthetic A-beta oligomers, which points out that A-beta oligomers are pathogenic synaptic ligands. So these types of experiments, and by the way, this diagram from the NIH Progress Report on Alzheimer's disease was changed after our discoveries to include not just amyloid plaques, but in fact, to attribute neuron damage to A-beta oligomers. And this neuron that you see as damaged here was the same neuron that you saw in the previous slide, which was used as a model by the artist at the NIH. And these little gold spots here are the oligomers targeting synapses on the damaged neuron. At NIH, Eliezer Maslia, who's the director of neuroscience for the National Institute on Aging, has concluded that progressive accumulation of A-beta oligomers has been identified as one of the central toxic events in Alzheimer's disease. Or put another way by Dave Teplow, one of the originators, in fact, of the original amyloid cascade, wrote that the oligomer cascade hypothesis, which states that oligomers are the initiating pathologic agents in Alzheimer's disease, has all but supplanted the amyloid cascade hypothesis. So if we're thinking in terms of next generation Alzheimer's immunotherapy, the goal would be to, in fact, target A-beta oligomers, not the plaques. And there has been progress at this level at Acumen, in fact, in an 8-year research and development program that led to the antibody Acu-193, which is a humanized A-beta oligomer antibody, an IgG. What you're looking at here is this 5xFAD transgenic model that I said develops memory problems when it develops oligomers, and the model developed by Bob Vassar. If we do the novel exploration task, the wild type loves the new, but the 5xFAD can't tell the difference unless it's injected with Acu-193. So post-injection, the 5xFAD, same ones that couldn't remember it here, now are very good at discriminating what is new from what is old. So to get back to Biogen's antibody, which has a bit of efficacy, not as robust as we would hope, and some people don't think the evidence is strong at all, but the target of aducanumab is oligomers, but also amyloid fibrils. And the antibody at Biogen was not developed to specifically target oligomers, but the Acumen antibody was. So the Acumen antibody doesn't bind monomers like Eli Lilly's antibody did, solanezumab. Aducanumab binds quite well to plaques as well as oligomers. Acumen is antibody is specific for oligomers, and as you just saw, is very effective at rescuing memory. And of course, all of the other preclinical work has been done as well, which led to IND approval for Acu-193 in April. And in fact, the first clinical trial steps are active as we speak today. So Alzheimer's patient Augusta D was covered very thoroughly by Alzheimer in his neurology workup notes. And one of his pages referred to the time he asked Augusta D to pick up a pen and write down her name. And she stared at the paper and couldn't do it. And what she said is, "I have lost myself." And that, in essence, is what's going on in Alzheimer's disease. And we see the trajectory through the disease by American artist William Udermoens to give us a sense of what it must be like to lose yourself. He was diagnosed with Alzheimer's disease 5 years before this. So what I've said is that A-beta oligomers are neurotoxins found in the AD brain, and I would guess that they were present in both of these individuals. And Alzheimer's disease is going to be a tremendous problem in the near future, estimated to be $1 trillion a year in 2050. I believe the new A-beta oligomer antibodies, such as Acumen's or perhaps others, may help avert this crisis. And in the long term, we also might look toward vaccines and small organic molecules. I do need to, since we've worked in this field for a long time, I need to thank our very many collaborators and also give special thanks to our terrific teams over the years and for a great deal of support. So thank you very much.
Wonderful, Bill. Thank you so much for that. And we've got maybe 2 questions we can knock out here. And one's from Jay, where he's asking about control brains in some cases have more A-beta deposition. Why then don't you see oligomer staining? Is A-beta in these controls not in their oligomeric form?
Right. So in fact, the plaques might be a storage depot. You could speculate. Storage depot for the different forms that are needed to create oligomers. And if you look at a transgenic mouse, for example, what you see is oligomer levels go up very early. We did some work with Frank Lafurla out at UC Irvine with a 3X mouse. Oligomers go up early, then they go down as plaques form. And then as the plaque levels saturate, the oligomers start to show up again. One has to look pretty closely at the immunohistochemistry to really establish what is the case, though.
Mm-hmm. Another one from Dean here is the enrichment of A-beta oligomers in AD patients CSF from the 2014 paper looked impressive. Has this observation been repeated in other studies? And what is the extent of the uptake of this biomarker for patient selection, stratification of disease progression?
Great question. And in fact, Dennis Elko and his team at Harvard have tried to set up the assay. It's extremely difficult because the levels, in fact, are quite low. And I don't think the assays that you typically see in the literature now are appropriate. Chad's assay is quite difficult to run, and it took over a year to get those data. So the results are clear cut, but as a day-by-day assay, it's not ready for prime time.
Yeah, and so this isn't a question as much as a comment from Vank about he'd love to hear you opine on the need for practical and rapid intermediate outcomes to determine clinical efficacy in the development pipeline. So we'll have to skip your opining there. I think due to time, I would like to hear that as well. Maybe we'll end. So Badir and Ezra also had questions, one about exosomes. Maybe we can pick that up later in the program. That's an interesting angle. But why don't we end with Badir's here about, do A beta oligomers increase overall tau level or just tau phosphorylation? Do these oligomers kill neurons irrespective of what method you use to differentiate? So maybe just the tau, how does it relate to tau and phospho-tau? Yeah.
Right. So, in fact, I mentioned this work we did with Frank Furla a while ago. And in fact, if you come into the 3X mouse, the triple transgenic mouse, it develops tau pathology. But if you use, in this case, it was an old polyclonal from their laboratories. If you clear out oligomers with this polyclonal, you also get rid of the total tau pathology, which was elevated.
Yeah.
So that was the answer to that. Great.
Well, maybe we'll sneak one more in about, what about a combo antibody approach for tau and A beta? Has anybody proposed that?
Well, I think combination therapy is a hot topic, and it's certainly likely to be desirable, and you might even be able to find a single antibody, like the notion that Hande was talking about, try to find something that can target more than one mechanism at the same time. One could perhaps develop an antibody against oligomers of tau and oligomers of A beta and get rid of them together, which would be terrific. And that actually goes back to the exosome question too, which we could come back to later.
Great. Well, Bill, thank you so much for that. And boy, we're rolling. And boy, the Q&A is hot. There are lots of questions coming in, but we'll have to move on now to Genia, who is the Soretta and Henry Shapiro Research Professor in Molecular Biology here and Associate Professor of Neurobiology in the Weinberg College of Arts and Sciences here at Northwestern. Go Cats and go Genia.
Hi, everybody. Thrilled to be here to have a chance to talk to you today about neuroplasticity. So this is primarily with a strongly pretty preclinical focus for our lab. Let's see if we can move the screen forward. All right, so your brain can be thought of in some ways as this kind of a electronic schematic. There are billions of neurons that are wired together into complex circuits by literally trillions of synapses that have been already introduced today, the tiny junctions that neurons use to communicate. But the real brain is different in a few really important ways, and you could distill it down to two aspects. One is that these synapses are continually changing. They're really dynamic. They are created and unmade throughout your lifetime, not even just as a function of disease. And the other really important difference is that overlaid on the electronic-like fast neurotransmission circuits of the brain is a rich array of different kinds of neuromodulators that have been linked to many neurodevelopmental, neurodegenerative, and mental health disorders as well. So the broad directions of our lab is to try to understand how neuromodulation controls the plasticity of neuronal connections. And I'll show you some really pretty exciting essays we have developed that facilitate this work, and how different kinds of neuromodulatory systems interact with one another in order to control brain structure and brain function, because of course the two are very tightly related. And finally, about sort of a third to sometimes up to a half of the lab is dedicated to developing new kinds of tools. And that includes tools for genetically targeted proteomics, better imaging of neuronal structure and function, as well as an interesting new screen that I will introduce to you today. So neurons are very, very different in our brains. This is true for us. This is also true in mice, our animal model of choice. This is a very classical picture from Ramon y Cajal, the father of neuronal doctrine, who has made the world recognize that neurons aren't just the syncytium of interconnected canals in the brain, but rather they're discrete cells that sometimes have really complex dendritic structure, at other times very simple, and aspects of the structure and connectivity is enormously important to determining how they function. This is a somewhat more modern view of a neuron in a living brain slice from a mouse. And in this case, we're not using a kind of a textile dye, which is how neurons used to be visualized, but rather a genetically encoded fluorescent protein that allows us exquisite specificity for looking at different kinds of neurons in the mouse brain and in different states. And in addition to the cell soma and the dendritic tree, I want to draw your attention to dendritic spines that Bill has already introduced, these tiny little dots that stud dendritic arborizations. So these are membrane protrusions that compartmentalize many, although not all, excitatory synapses for the majority of neurons in the brain. And this is their turnover and their dynamics are one of the kind of a core feature of how the vertebrate brain operates. So if you look at a little closely at the dendrite, you see a lot of these dendritic spines that are quite different in their shape and size, and they all protrude from what we call dendritic shaft. Typically, dendritic spines correspond in essentially a one-to-one way to an excitatory synapse. And this is fantastic because it gives us a morphological traction over connectivity levels. As I had already mentioned, they change in their size and shape, and they also change in numbers, not just in neurodegenerative states. So how do we typically study this? We can look at dendritic spine density and the shape of individual spines. And we use that using different kinds of imaging approaches. And there's a lot of different downsides that are associated with these imaging-based metrics. So the loss of this normal turnover of dendritic spines and the synapses contained within is typical, of course, for neurodegeneration, often being one of the earliest biomarkers of changes that will later cascade towards the death of a neuron. There are many neurodevelopmental conditions that are associated with miswired circuits where you see some changes in plasticity. Mental health disorders have also been linked to it, and even to some extent normal aging. So the potential, however you look at it, dendritic spines and synapses hold a really key aspect of neuroplasticity, and almost any kind of disease you care about that involves any aspect of neurology or mental health. And there's going to be either a root cause or a really crucial correlate in the readout for neuroplasticity. So it's very important to have good ones. I will show you a really unique assay next, where we're able to causally induce the formation of a new dendritic spine at will, basically. So if you look at this location in the image, we will cause a new spine to grow there. Give it just a second. And there it goes. I'll see if I can play that again because it's pretty quick. This is sped up about twofold, so the process is really fast. And we think that these kinds of causal plasticity induction is a really powerful way to study how different kinds of molecules alter the potential for plasticity on neurons. In this case, neurons that are in an acute brain slice, so with strong genetic targeting and within a relatively realistic circuit context. So how do we make new spines grow at will? We use lasers in order to do that. So this is an interesting chemical which contains a caging group, like a tail, that's tagging a glutamate molecule. And this bond here is light sensitive. So if we shine a laser of a certain wavelength on this molecule, it'll fall apart into the inner tail and the molecule of glutamate. And in the brain, glutamate release is an important causal factor for the formation of new synapses and spines. So if you do that while you record from a neuron, you can literally find the actual synapse, release glutamate there, and see how strong is this particular synapse. And a few years back, we realized something quite extraordinary. If you don't record from this neuron, which washes out really important plasticity cascades, and instead uncage glutamate near a dendritic shaft that's expressing a green fluorescent protein, for example, something that allows you to visualize it with another laser that's tuned to an imaging wavelength, you can actually induce the formation of a new dendritic spine. And this is an assay with very high spatiotemporal control. It only takes a few seconds to cause or not cause probabilistically the formation of a new spine and synapse. This has exquisite cell type specificity in the mouse brain, and you can use it within a relatively normal circuit context. And these new dendritic spines come to carry functional synapses, which we can show by doing a fairly complex experiment where you seal onto a neuron, but not break into it in order to record from it, induce a new spine, and then try to gain electrophysiological control over the cell. And then you can show that, look, this new spine has an actual synaptic current, smaller synaptic current than its older neighbor right here, but nevertheless, within a matter of minutes, aggregating a kind of a protosynapse. Okay, and this is just a comparison of a few of these nascent lab-grown and pre-existing spines. And so we think that this is a really spectacular kind of assay that causally allows you to test the potential, the capability for plasticity that can be used with many different kinds of drugs. And we've recently used this approach in order to make some surprising discoveries about a drug, ketamine, that appears to work as a rapidly acting and temporary antidepressant, whose mechanisms is not yet completely understood. And we've discovered that ketamine's effect on plasticity apparently works via recovery of dopaminergic signaling. And so to do these kinds of experiments, one can target a particular neuronal population of interest with a virally transduced green fluorescent protein. Then you can prepare acute brain slices and carry out a lot of these induction attempts so that you get an aggregate potential for probability. So this is another example of a successful spinogenesis and an unsuccessful attempt where clearly no new spine was produced. And we were able to do this over and over again and determine that ketamine, in fact, co-opts dopamine receptor D1 mediated cascade acting through protein kinase A in order to recover normal levels of plasticity in medial prefrontal cortex in a way that appears to relate to behavioral improvement in models of aspects of major depressive disorder. And so this is a kind of a precise and causal moderate throughput readout that's compatible with behavioral manipulations, with really intensive pharmacological dissection of mechanisms as well as genetic ones. And so we think that that is in many ways superior to kind of a snapshot readouts of changes in overall dendritic spine density, shape, and size. And yet, nevertheless, we still need much higher and better assays and screens for tracking neuroplasticity. So let's look at what we've got so far as a field. There are a lot of really good non-neuronal high throughput assays. They're basically, by and large, irrelevant for screening for neuroplasticity at dendritic spines and synapses. The cells are just too different. They don't really work the way that neurons do. Neurons are very specialized cell. Dendritic spine counts are a laborious endeavor, relatively error prone, and allows you to see snapshots of the brains at different time points and not continuously. Snapshots that also cannot really dissociate changes in spine turnover from spine production. In vivo imaging assays exist. That's very labor intensive and has limited pharmacological traction. And finally, there's this new glutamate uncaging way of causally interrogating plasticity that's kind of intermediate in terms of a labor investment and is a direct readout of plasticity that's quite powerful, but not as fast as we would ideally like. So what do we want? We want something that can get us away from imaging. So something that tells us about how plastic are these dendritic spines and the synapses contained within, but without having to image them. That would be the fastest thing that we could have. And well, since an assay like that didn't really exist, we had to go and create one. So for this part of the work that I'm talking about, the provisional patent has been filed with our Northwestern in vivo office. And so a graduate student and a postdoc in the lab had thought that perhaps we could target structural plasticity compartments and leverage an enzymatic readout so that we can get away from this laborious imaging approach. And so we created a series of constructs where a part of a construct leads you to compartments which are potentiated, that have recently undergone a lot of activity. And another part of the construct makes sure that you're located at excitatory synapses contained in dendritic spines. And yet another part of the construct makes sure that you don't mess up the functioning of those synapses. And now what we can do is use a large scale primary neuronal culture system, so real neurons that actually have spines and synapses. And then we can screen a lot of ligands with an enzymatic readout via luciferase. So this is kind of an example of a luciferase signaling that can be readily read out with a machine without necessarily having to form an image. And so here's an example of some of the earliest experiments we have done with this approach. First, just a screen of our 3 different constructs, and no luciferase control gives you no luminescence, and forskolin, a known plasticity inducing agent, gives you kind of a sigmoid-like curve for the 3 constructs, with one being clearly a stronger induction than the rest. And then we can move to primary mouse neuron culture and then examine again a bunch of different kinds of positive controls of compound, including ketamine, which we study, that seem to activate the plasticity of dendritic spines, as was shown by our uncaging evoked screens. And you can see that all of these different drugs, except for the DMSO control, are associated with an enhanced luminescence readout. And then in one final pilot, we also showed that we're able, for a particular plasticity-inducing compound, we can block a large fraction of that induction of plasticity by using a downstream inhibitor of some of the signaling cascades that are activated by this particular compound. And so the screen seems to work. I think it's very well applied for starters for large-scale screening using mouse neuron cell culture, because it turns out that many of the crucial types of signaling cascades tied to plasticity are actually conserved between mice and humans. But we think that the potential is greater, and you could imagine using differentiated pluripotent stem cells for more personalized medical applications in the future as well. And of course, you can then validate, and we plan to validate the top hits from the screen using these kinds of more targeted functional plasticity interrogation efforts that we can do in our lab. So in sum, this is a brief update on the state of neuroplasticity of dendritic spine interrogation. We've created some, we think, fairly efficient screens for screening neuroplasticity effects using drug libraries and for new compounds. It's a really critical and currently unmet need because of the limitations of the imaging assays I've described. We think that this causal fancy laser-based method for inducing new dendritic spines and synapses is quite a useful new method to assay plasticity potential for the top hit compounds that pass certain prior qualifications. Enzymatic approaches offer scale-up potential that will never be matched by imaging, at least not anything that we can attain in current state, no matter how much AI-based approaches we throw at automatic classification. And here we can leverage the wonderful powers of the high throughput analysis laboratory, which is one of the many crucial CLP cores, which gives us access to well over 100,000 compounds across several crucial libraries that we hope to bring into our screen as soon as possible. And with that, I want to acknowledge my lab, our various funding over the years, and some of the key people behind the work that I talked about today. Vas came up with the screen, very much helped along by Pushpa Kumari, who's a postdoc in the lab. Most of the uncaging evoked spinogenesis work is from our spine and synapse whisperer, Mingzheng Wu. And with that, I hope I have time for a little questions.
You do, Jenny. Thank you so much. That was great. And here's one of them. How does your reporter differ from other activity reporters like FOSS, KREB, ARC, Campari?
Well, we're targeting, I mean, it's similar. There's some subtle differences in the exact promoter sequences we're using, and we are trying to target multiple of these transcription factors. So we're never going to settle on a single kind of construct, and instead we're planning to create about 4 or 5 that perform really well, that are based on different kinds of transcription factors. And then the other difference is the targeting, right? We put it at the synapse so that our enzymatic activity is tightly linked to what happens directly at the synapse, not in the cell body, where a lot of these transcription factors have also really important functions as well that are distinct.
Yeah, you kind of, at the end there, you described use of the assay in a disease model. You know, which which one might you favor, or what rises to the top in terms of the most viable or the most exciting from your side? Which disease model would you try?
Well, it depends on who gives us the money to do it. To some
Okay,
extent.
all right.
But I think I'm really excited about, of course, trying some cases where we have some genetic traction, right? Where there's a gene or else a couple of genes that we can modify so that you can create cultures that carry the genetic mutation and cultures that don't, and then really rigorously assay the potential efficacy of different kinds of compounds.
Yeah, well, so right after, I see there's a lot of interest in that. So anonymous attendee asks, can you do a genetic screen to actually try and link the genetic abnormalities to your screen for neuroplasticity?
Absolutely. So you can do that by creating and then correcting different kinds of genetic abnormalities in your culture model. Something else that we're really excited about, I didn't have time to talk about our proteomics work in this talk, but we also want to understand how the proteins at the synapse are changing, because we know that multiple signaling cascades converge on regulating plasticity. And so we hope that we may be able to kind of combine proteomic level insights with interrogation of plasticity, with genetic perspectives, in order to understand better some of the changes that are occurring in different neurodegenerative and neurodevelopmental disorders.
Yeah, I've had a front row seat to that in your spatial proteomics, very localized, cell-specific, it's pretty amazing to see that kind of advance, biochemical level, protein level information right there where it counts. Yeah, maybe next time we can get to that. Maybe a last one here. Could you comment further on the iPSC derived models and potential functional genomic screens like CRISPR and RNAi?
Yeah, I mean, I'm very excited with iPSC driven technology, because we can then basically you could basically screen drugs for an individual patient. And this is something that you heard already a lot about from Hande. And I think our kinds of screens is a way to facilitate making that happen.
Great. Okay. Well, Jenny, thanks so much for that, and getting us up to speed on neuroplasticity. We're going to switch to our cleanup hitter, our fourth in the lineup today. Yep. Thanks again. And that's Rick Silverman. And he's the Patrick Ryan Aon Professor of Chemistry and Molecular Biosciences here in Weinberg School at Northwestern. And so without any further ado, I give you Rick Silverman.
Thanks, Neil. So there was a change in my title, as you may have noticed, because I realized in 15 minutes I couldn't talk about two projects in my group. So we're going to focus today on the development of a potential new drug we call NU9 for treatment of upper motor neuron diseases. And so this is sort of a continuation of what you heard about, I don't know, an hour ago or so from Hande Özdemir about upper motor neuron diseases. And our focus is on inhibition of protein aggregation. And so protein aggregation is really important to all the neurodegenerative diseases. We're going to focus today on amyotrophic lateral sclerosis, ALS. It also, upper motor neuron disease would include primary lateral sclerosis, hereditary spastic paraplegia as well. ALS is really a horrendous disease characterized by premature degeneration and death of motor neurons. And the patient generally dies within 3 to 5 years of diagnosis. Now, 10 to 15% of ALS is hereditary, and a quarter of the hereditary disease comes from mutations in an enzyme called superoxide dismutase 1, or SOD1. Mutation in SOD1 forms aggregates. They then accumulate in the neurons and cause the neurodegeneration. There are only 2 FDA-approved drugs for ALS today, Riluzole and Edaravone. And so we're always looking for new treatments, and particularly for these unmet medical needs. So for protein aggregation then, we set up 2 screens. These were PC12 cells expressing mutant SOD1. We looked for aggregation as a result of the mutation, and then the cytotoxicity that results from this aggregation. And we ran a high throughput screen, greater than 50,000 structures were involved. And this led to quite a number of hits that we could put into various structural series. There were 17 hit series we identified. We then filtered these series with ligand-based computational methods, substructure similarity searching, iterative clustering, and we honed that down to series that then seemed most promising. We then ran a screen through the Zinc database, which is, well, at that time it was only about 2 million compounds. Now it's got to be, I don't know, 50 million commercially available compounds. But we ran through to see what compounds would have similar properties that we could then test. And so based on those studies and medicinal chemistry review of these compounds, we came down with 3 lead series. I'm not going to have time to talk about the 3 lead series. We're going to focus just on one of the 3 because that's the one that led to the compound NU9. And so this series has this structure, cyclohexane 1,3 diones. There were about 50 compounds in that screen that did show activity. Unfortunately, I can't then spend much time telling you how we modified that initial structure, but after about 5 years of optimization, we eventually came upon a structure of this sort that was penetrable in cortical neurons. It's a chiral molecule, has 2 asymmetric, has an asymmetric center. So we synthesized each separately and found that the S isomer was in fact the one that contained most of the activity, and that molecule is NU9. For the organic chemists out there in the audience, I thought I'd throw a synthesis slide in to show how easy it is to synthesize this molecule, starting from the R isomer of ethyl lactate, doing a Mitsunobu inversion to convert that to the S stereochemistry. We then reduce the ester with Diebol to the aldehyde, condensed in this Wittig reaction to produce this conjugate. This then was condensed with diethylmalonate to produce that cyclohexane dione ring. Hydrolysis of the ester followed by decarboxylation gave NU9. And just this past week, actually, I was told that our 1 kilogram of NU9 that I ordered from a CRO is ready. It took them about 6 weeks to prepare a kilogram of this. So we will be starting a lot more animal studies with that. The compound has great pharmacokinetics, ADME, its plasma and microsome stability good. Blood levels remain high for a considerable period of time, penetrates the brain well, and it's orally bioavailable, 94% orally bioavailable. So this will be an orally administered molecule, as you'll see. It does cross through membranes well with very little efflux out of the brain. So once it gets into the brain, it doesn't get shot out of the brain. Little or no metabolites are produced from this molecule, so you don't have concerns with metabolites that could cause issues. Off-target properties are good. It doesn't inhibits cytochrome P450s. These are metabolizing enzymes that if you interfere with them, they can cause drug-drug interactions, and U9 does not. The hERG channel is a potassium channel in the heart that regulates the beating of the heart. You don't want to mess with that one, and our molecule doesn't. We then did some safety study, toxicology studies, and a single ascending dose study in mice went up to 1,200 mg per kg. This is in a mouse. If this were a human, 1,200 mg per kg would be like 8 or 9 grams of this a day. And it showed no adverse effects and no external or internal abnormalities. When we carried out the 7-day repeat study, however, after about 2 days, there were some adverse effects that showed up. By the time we got down to 300 mg per kg, these adverse effects were really minor. But when you got to 100 mg per kg per day, you reached the no observed adverse effect level. So these mice looked identical to the wild type healthy animals. This is active in the ALS mouse assay. It extends the life of the ALS mouse. However, an hour ago, we were told by Hande that that mouse model is useless because it really doesn't translate to humans. And so it almost seems ridiculous to do an assay that there is no indication as to whether it's going to be useful in humans. And what she has found in her studies is that it's the corticospinal or the upper motor neurons that start to degenerate early in this disease. And if we could maintain the health of the upper motor neurons, then that should then translate to humans, because these upper motor neurons are structurally and functionally quite similar between mice and humans. And this could be a new paradigm then for identifying molecules that would translate from mice to humans if we could show the effect, beneficial effect on upper motor neurons. And she told you how she did it. This is amazing. There are like a couple billion neurons and only a couple 100,000 upper motor neurons. But she was able to selectively fluorescently label the upper motor neurons by recognizing that there's an enzyme in upper motor neurons that's highly expressed. It's not expressed well at all in other neurons. And so she was able then to fluorescently label the upper motor neurons exclusively. And those labeled upper motor neurons were identical to the ones that weren't labeled. She then crossed them with ALS mice, and she started out with the SOD1 mutant mouse, and then also the TDP43 mutation. She's now has done Olson and Perfilin and spastin. And so she has quite a number of these mice that are crossed with the fluorescently labeled mouse. So now you have ALS mice that have fluorescently labeled upper motor neurons. And so now we can look at the health of the upper motor neurons in mice. And since they are really quite similar in humans, this hopefully would translate to humans. So let me tell you about the study she carried out in her laboratory. These are all studies from Hande's laboratory using NU9. So, and these are all orally administered. So these studies were done by oral gavage. You give the compound in solution to the animal. And the study doesn't start until the animal is 60 days old. This is when the ALS mouse starts to show symptoms of ALS. Of course, you don't want to give a drug in a preclinical study before a human would even know they had the disease. But now these are mice where you know they have the disease because they're starting to degenerate. And so this is when the drug is administered. And just looking at the general upper motor neuron health, here is the healthy upper motor neurons of the healthy mice, the wild type mice. And these are the mutant mice. You can see very few of their upper motor neurons are healthy. If you administer at P60, 20 mg per kg or 100 mg per kg of NU9, you see that the upper motor neuron health now is pretty comparable to the wild type animals. Apical dendrites start to form vacuoles when they're degenerating. And so you look at how many vacuoles are formed, and in a healthy mouse, there are relatively few vacuoles, but the mutant mice have a lot of vacuoles. You treat with 20 mg per kg, 100 mg per kg, and now we're down at the level of the healthy animals. Misfolded SOD1 proteins, the wild type, they're very few. The mutant mice have a lot of misfolded proteins, 20 mg per kg, 100 mg per kg. You can see that there are many fewer misfolded proteins. Axons are really important in the health of the upper motor neuron, and you measure this often with axon length. So the wild type animals have axon lengths here, about average 320, 330 microns. The mutant animals have much shorter axon length. This is the treatment with NU9, comparable or longer actually than the wild type. And here are the two FDA approved drugs, Realizul and edaravone. They aren't nearly as long as with NU9. We've heard that mitochondria is really important, the endoplasmic reticulum. Here's a healthy mitochondria endoplasmic reticulum. Here's the mutant animals. The mitochondria is almost empty, and the ER is a mess. Here's treatment with 100 mg per kg. Looks a lot like the healthy, and the ER looks like the healthy. And there's a graph to show it. This is the percent of healthy mitochondria in a wild type animal. Here's a mutant animal, and then here's treated with 100 mg per kg of NU9. So all these studies were done with the SOD1 mutant animal. That was the animal that we, or the mutation we used in our high throughput screen to identify eventually NU9. But Hande had, as I said, other mutant animals. And so she carried out the same study using the mice that had a mutation in TDP43, which is totally unrelated to SOD1, and got the same result. And so here again, this is healthy mitochondria of a healthy animal, the TDP43 mutation, and treatment with NU9. Same with axon length. There's the healthy animal, the mutant animal length, and here is NU9 length, quite large, whereas Realizul and edaravone much smaller. Upper apical dendrite with TDP43. Here's the wild type. Here is the mutant one. We have many more vacuoles. Treatment with NU9, 100 mg per kg, we have very few vacuoles. And the overall upper motor neuron health of wild type compared to the mutation, and then 100 mg per kg NU9 brings it back to the wild type. Body weight tells you something about the health of the animal, and the non-mutated animals usually weigh more than the mutant ones. But the point is, if you give a drug to either the healthy or the mutant animals, the body weight doesn't change. So they stay the same throughout this entire study. Same with the SOD1 or the TDP43. And then finally, a behavioral study. You have this wire mesh that the animal grabs onto, and then you invert it. So the animal has to hold on to this mesh, and you're measuring the grip strength. How long can the animal hold on. And you see that after a period of time, the mutant animals start losing their grip strength and they hold on less and less. But the animals that are treated with 100 mg per kg of NU9, this SOD1 or the TDP43, they maintain their grip strength. And so we started out with the screen, just looking for inhibitors of protein aggregation based on mutation of SOD1, and eventually came up with this molecule NU9, maintains the healthy upper motor neurons, both in mutant SOD1 and TDP43. And so we think this could be a paradigm shift in how you identify molecules that might translate from mice to humans in upper motor neuron diseases. We need to try the other mice with mutations that maybe this drug would only work for these two mutations and not others. And then you can use this, as Handi says, in personalized medicine to identify which people would get this kind of drug. So it took quite a number of people to carry out this study. And I thank all of them for their contributions, and thank you all for listening in, and ready for some questions.
All right, Rick, thank you so much for that. There is the first one here from an anonymous attendee asking, how does your drug approach compare to the new CRISPR G93A corrections? For example, Chen-Yun Lee's group.
Yeah, well, I mean, mine is a small molecule, orally available small molecule. We're inhibiting the protein aggregation. We know the target is the upper motor neurons, but we don't know what is the actual protein target. You need to know that for CRISPR, so you know what to fix. And so at this point, we don't know what to fix. All we know is that this is maintaining the health of the upper motor neurons.
Stephen asks, what is the status of NU9 development, I think, in terms of translation, and what stages have you brought it to?
Yeah, so we are currently doing IND enabling studies. I say I got 1 kilogram of this compound to get going on the toxicology, safety pharmacology. I would guess this could be 18, maybe 24 months before FDA approval, if it does follow through as well as we hope it does. And so clinical trials could be that far away.
Are you prepared to disclose who's carrying out the pre-IND studies?
Me, my company.
Aha.
So this is Akava Therapeutics. And so we're starting to do those studies.
Great. And another question about, does it work in the TDP mouse model? Does NU9 work there? Have similar effects in broader ALS subtypes?
Yeah, so the last several slides there were the same studies we did in SOD1 mice that we then repeated in the TDP43 mice. And so, yes, it works in both animals. That question probably came up before I got to that part in the talk.
Oh, yeah, okay. And so Nick asks, are there any synaptic electrophysiological deficits in these ALS models?
I'm going to leave that up to Hande to respond if...
Yeah, I can respond. We actually published a Frontiers paper. There are electrophysiological defects, and now we actually purchased an equipment which will allow us to do mapping studies and to see at what stage and level those connectivity defects occur and how NU9 improves that.
Okay, great. Well, there's some interest, and so I think any of the speakers, for those interested, absolutely direct email to the speakers. They're the masters of their domains, and I encourage those with questions about follow-up issues to approach them directly. And that concludes our Northwestern faculty presentations. Thanks to Hande, Bill, Jenny, and Rick for taking the time to join us. And I hope you see in the Midwest, we underpromise and overdeliver. Next up is a distinguished group of experts who'll discuss hot topics in neurological disorder therapeutics in a panel format, and that's guided by Lauren Martz. And so I will just directly hand it over to Lauren, please.
All right, thank you, Neil, and welcome everyone to our discussion on hot topics in neurological disorder therapeutics, which with the approval of aducanumab, I think couldn't come at a better time. I'd like to introduce the panelists joining me for the discussion. We have Sarah Bagat, General Partner at Sofinova Investments, Ginger Johnson, CEO at Cello Health Bioconsulting, Norbert Riedel, CEO of Aptinix Inc., and Mi-Young Shin, Executive Director and Head of Early Discovery Genetics and Pharmacogenomics at Merck. Like the other sessions, we'll take some questions at the end, so feel free to use the Q&A function to ask questions. Okay, do we have the panelists on here? I see we have Sarah,
yeah.
so
Norbert is also
and
here.
Norbert, great.
Yeah, I'm here too, Ginger.
Okay, perfect. All right, Sarah, I will start with you. So as an investor, I'm wondering, what do you see as the implications of FDA's decision to grant the accelerated approval for aducanumab for other companies working in the CNS space?
Well, I think first and foremost, there's obviously a huge unmet need here with Alzheimer's. There's, roughly 35 million patients in the world who have this disease, and we really are in need of therapies. And it's great, on the one hand, that the FDA is sort of working with the industry to be able to create kind of biomarkers to be able to get these drugs approved so that there is something for these patients. I think the challenge is verdict's still out, whether levels of amyloid plaque are correlated to disease at all. So on the one hand, I think it's great that we're trying to create ways to measure success for some of these therapies early on to sort of innovate more development, more drug development, and more focus in a space with such an unmet need. The challenge is, do they essentially stimulate development of drugs that impact amyloid levels in the brain over other mechanisms that may be more beneficial? So I do think that there's, and without clarity of really understanding the phase kind of 4 post-marketing studies that will sort of hinge this development over the next 9 years with Biogen's drug, I just think we could be in a challenging place with essentially stimulation for development over this mechanism over others. So I don't know if this is necessarily an overall good outcome. I think there wasn't really enough data to really make this decision, although I appreciate the need for a more kind of customized path forward for a lot of these therapies to go through. So I think with more collaboration with the FDA, hopefully there are more a stimulation for more drugs in this space. But we really have to focus on biomarkers that are going to be correlated with clinical benefit. Alzheimer's is a slow progressing heterogeneous disease. Clearly it takes, before a patient even has mild symptoms, they've been progressing for decades. And so trying to figure out what are the biomarkers that are most sensitive, objective, and clearly correlated to disease are going to be very important. And so I hope there is more collaboration moving forward.
Sure. And I guess when you're looking at companies working in the Alzheimer's space specifically, are there certain biomarkers that you're looking at that you think might be more promising than amyloid beta levels?
I think that's the question, right? That's the, I guess, billion-trillion-dollar question. It's really, what are the biomarkers that are correlated with disease? And I think, unfortunately, we don't have those answers yet. And so I'm hoping that there's more clinical research, but it probably will take a significant amount of time because it is slow progressing to try to, really identify what are the biomarkers that are most meaningful. How do we measure what level of difference is going to be beneficial to these patients? What are some of the other metrics and other cognitive scores that are pretty kind of reliable and validated? So I don't know, beyond just seeing the existing path, and I don't think first, I think there are a lot of people who are not convinced that amyloid beta is a biomarker that's really going to read out for a clinical benefit. So I'm not sure it's going to stimulate, other than people who will just, I guess, in terms of there is now a path forward, right, for this mechanism. So that will stimulate, I'm sure, more funding for those products. But I'd still like to see stronger data before, I personally would be convinced that additional funding for similar products would make sense.
Okay, that's a good point. Norbert, I'd like to ask about other areas of unmet need. I think, Alzheimer's is still certainly a huge one, but what other areas need a lot more focus in the neuroscience space?
Look, to me, it cuts across areas of neurodegeneration more broadly, Alzheimer's disease being one, but there are numerous other diseases that have cognitive impairment and dementia, neuropsychiatry, in particular areas like PTSD. I mean, we heard earlier, right, that before this approval that Biogen achieved, there hadn't been anything approved in 20 years. In neuropsychiatry and PTSD, we have 2 FDA-approved therapies. They are both SSRIs. They were approved more than 20 years ago. And in my view, they offer some symptomatic relief, in particular in areas of depression associated with PTSD. But fundamentally, when you ask the question, do we have anything today available to patients, millions of patients, in these areas of high unmet need? I think the answer is generally no, not really, because the therapeutic benefits are rather marginal. The side effects are, in many cases, not particularly suitable for chronic long-term treatment of these disease states. And having said all of that, and of course, listening to the presentations this morning, where we look at a way of resolution of individual sort of like brain components all the way down to individual synapses, and the discussion we just had with Rick Silverman on NU9, I think there are numerous ways now to have a much better understanding of the brain itself, plasticity changes, what triggers these disease states that we are looking at. Professor Klein mentioned, of course, the oligomers. And so I have high hope and high expectations that CNS is now more ready for prime time. I look at our own pipeline at Aptinyx and look at the results we have in several phase 2 clinical trials in chronic pain, which has a very central component in PTSD, as well as in Parkinson's disease. And I think there is a lot of promising activity that hopefully we can take all the way to the finish line and make a difference to patients that are in desperate need for truly effective and efficacious therapies.
Okay, great, thank you. Ginger, when we're talking about hot topics, I think we have to discuss precision medicine. So, I know this is an area that's taken off in cancer. And what can you tell me about the applications for precision medicine in neurology and the progress that's being made?
Been a lot of progress in precision medicine. The most high profile, obviously, is SMA with gene therapy. And I think, not to beat a dead horse, but if you think about the aducanumab and the approval there, the accelerated broad label, biomarker based, how that might translate to precision medicine, to me is very important. If you think of biomarker driven development strategies for genetic neurological diseases, most are rare diseases. You think, we've heard a lot of this morning, SOD1 expression in ALS, HTT expression in Huntington's. These offer a much more direct link between disease biology and clinical benefit. So if the FDA is willing to be a bit flexible and approve a drug on an accelerated biomarker-driven plan, and then look for post-market real-world evidence, perhaps there's a lower initial regulatory risk, and that the post-market studies can then be paid for with revenue generated by the drug once on the market. So what I'm hoping, thinking about what Sarah said, is that investors may see these neurological diseases, particularly those with precision, those that are focused on precision medicine and have real biomarkers that are connected to clinical benefit, as a bit lower risk. Neurology or CNS in general is never going to be low risk, but lower is a good thing, and shift some of that mountain of money in the biotech environment right now into neurological diseases, particularly precision medicine approaches.
Okay, and so I guess I'll direct this to Mi-Young then. I think with precision medicine, a lot of times we also think about new modalities. So I guess, what impact do you think that new modalities are having in neurology now?
So thank you. I think the relate to the previous question, and I think someone asked at the end of Rick's talk as well. I think the new modalities is up and coming. I think there's a lot of excitement over it. As you know, a number of companies have been partnering in gene therapy. Alnylam is very active in CNS for RNAi, and really I think opening up targets that we were not able to target by small molecule or monoclonal antibody. So, related to precision medicine, it allows you to go after some of these targets that were not tractable based on genetic findings. And for me, that's a potentially exciting avenue. I would say those technologies still need to be proven, a lot to be worked out. I think gene therapy, certainly in other areas, have been in the clinic, has not been very successful. I would say with limited success, and the complexity of the heterogeneity of disease that's been discussed, along with the complexity of the human brain. I think, you really have to figure out how you're actually going to get to the specific cell types that you want to go after to actually deliver these and make the change that you want to do. But I think it's an exciting field. I think, certainly, a lot of companies are interested in it. What the time frame of those modalities and how efficacious they will be, I think is still a question mark.
So it sounds as though delivery is certainly one of the biggest challenges when you're looking at new modalities. Are there other challenges that need to be overcome to implement things like new modalities or precision medicine in the neuroscience space?
Yeah, I think certainly with genetic manipulation like CRISPR and others, you do have to worry about potential. I think the technology is getting better. You could actually look at these off-target events much better, but I think there's still some concern with that. I think the whole moving away from DNA to RNA editing is exciting because you may actually get away from manipulating the DNA. I think the siRNA platform is pretty solid. So I think for them, it is more of the delivery question and how to get at it. Certainly they have knowledge shown that it could be pretty efficacious with very minimal side effects using siRNA.
Sure, and some of these genetically defined, subsets of certain neurological diseases seem to be a good testing ground for some, especially genetically targeted therapies and new modalities. What needs to happen to move these hot topic ideas into the more prevalent indications and patient populations within those diseases?
Yeah, I would go back to Hande's talk, right? I think she's, what she outlined is trying to understand what are the mechanisms and what are the pathways that are relevant for different human neurodegeneration disease or other neuropsychiatric disease, and try to stratify those. And if you could actually go show that those mechanisms that are relevant to rare disease is also relevant in common disease, then you may have a path forward expanding that indication. And I think to a certain level, I think, Rick's discussion of, the NU9 actually having an effect on mitochondrial biogenesis is exciting because I think there are a lot of the, potentially, we don't know what the targets are, whether that would translate, but I think, again, that potentially could be, again, a mechanism that may translate into much common disease. So I think that's where you're actually, if you understand a target and really the genetic basis for these, rare diseases and think that it will translate to much common disease, I think you'll be, where you want to move forward. Having said that, I think the Sanofi's pancreatic gives you a little caution as well, right? Because that certainly has been effective in Gaucher disease, but certainly going into Parkinson's patient it did not show to be efficacious, even though the underlying genetics people believe may be very similar in those two diseases.
Lauren, when you look at the prevalence of these diseases, there's like 10 million plus PTSD sufferers. There are millions and millions of Alzheimer's sufferers. I think ideally we want to understand a common denominator across these diseases so that we are not entirely focused on sort of like highly specific genetic variations, but more broadly think. And I was intrigued by Eugenia's presentation on measuring individual spines that are formed in the systems she uses. So anything we can do at the cellular level or brain slice level that validates a mechanistic approach to addressing neurodegeneration or neuropsychiatry, I think would be hugely beneficial and helpful. So we reduce the likelihood of advancing compounds into clinical development that are ill-defined or characterized and therefore have a very high probability of failing in clinical studies, because the translatability from a preclinical model to a clinical environment is often very limited. And the more we can validate a more sort of like common underlying mechanistic approach, I think the better our chances to also succeeding in the clinic.
Yeah, no, but if I may add to that, I think certainly that talk was exciting for me as well. I think from genetics point of view, the recent findings suggest that potentially synaptogenesis, spinogenesis pathways are very important, and I think marrying that genetic and the underlying mechanism, I think is going to be critical as we make breakthroughs in these diseases.
Yeah, I would agree with that wholeheartedly, and the opportunity is huge in that if you're looking for a common biology, let's say protein aggregation, right? You can address a myriad of diseases ranging from very small in patient populations to very large patient populations. And one of the challenges there is, where do you start? How do you select that first indication? And what are you trying to accomplish with that first indication? Is it proof of concept? And you never want to spend money just for proof of concept. You'd like to get a return on that investment. And what does it show you, and how translatable is it to the other diseases for that indication? And it's a good problem to have, but I agree.
Yeah, well, thank you, everyone. I think we just have a couple minutes left, so if anyone has a question, please feel free to put that in the channel. It looks like we have a couple here. So Michael Rosen is wondering how the COVID diagnostics, vaccines, and therapeutic development paradigm with the public-private partnerships and a massive amount of funding provide a future strategy for neurodegenerative diseases that have a really big societal impact.
Yeah, look, I think it shows you, right, the COVID diagnostic and the whole approach to a COVID vaccine shows you how powerful it can be to have these public-private partnerships and a global approach. I think we would all benefit from that being a concerted effort in CNS disorders. Having said that, I think it comes back to earlier comments we made, namely that there isn't a very obvious single target for any one of these diseases. The patient populations are very heterogeneous. The root causes are very, very different from one disease to another. And so I think it would have to be one that is focused on a broad array of new therapeutic approaches, a whole portfolio, if you will, of approaches to ultimately have a few that succeed and make it to the finish line. But clearly the power of of awareness, the power of funding, and an awareness of the societal impact of these diseases would be huge enablers for perhaps a stronger focus on funding and enabling these various new approaches we are working on.
I think the challenge is we still don't know what the finish line looks like. And I don't think the existing, aducanumab has really set that finish line with clarity. And so I think if we all understood where we're trying to guide and get to, could we do a more concerted effort with both public funding, private funding to get there? I think that then we could try to build off the COVID model. We still don't know what success looks like. We still don't have understanding kind of the right patient populations within Alzheimer's or any larger neurodegenerative disease. How do we measure success? What are some of these? How do we measure clinical benefit in a relatively short time frame? And for these diseases, 5 years is a relatively short time frame. How do you kind of fund that and test that appropriately? I think that's where we struggle. And then I think we could try to work better, with funding this and working with the FDA. But there's still a lot of basic questions that are not clearly understood.
Yeah.
Okay, and I think we'll take one more question here from Leland Gershel, is wondering about the small molecule for Alzheimer's disease, simufilam. I'm not actually familiar with that molecule, but it acts upon the scaffolding protein filamin A. And the question is, what do the panelists think about this approach and as a potential treatment for Alzheimer's disease?
I'm not familiar.
Well, I'm not familiar with the molecule itself either,
No.
but I would say certainly therapeutically, the small molecule will have a huge advantage over the antibody that's currently approved, in my opinion, right? I think the ease of delivery to the patients and certainly the cost of goods may actually be much better. So I think the small molecule for these mechanisms, if we could do it, is certainly still a way to go.
I agree, but we may be heading and probably heading to combination therapy, right? So you may have a monoclonal infusion and a small molecule and something else.
Sure, those are good points. Yeah. Well, I'd like to thank the four of you for joining me today and for a great discussion on hot topics in neurological diseases.
Thank you.
Thank you, Lauren. Thank you.
Great, Lauren. Thank you all and the panelists. And I'm going to pivot over to the corporate presentations now. But as I do that, as Sarah was just talking and listening to the Q&A, I'm personally invested in this because the measurements of proteins in proteinopathies is lacking. So as Sarah mentioned, many basic questions are open, and A beta 1 to 42, there's A beta 1 to 40. We've actually mapped 26 discrete molecular proteoforms of just A beta. And we're in the process of doing that for A beta oligomers as well, to be very precise about the proteoforms that are active. And, in astronomy, right? In the 16th century, we didn't know the sun was at the center of the solar system, but so we just measured the movements of the planets. We measured them very precisely, Tycho Brahe, and then 50 years later, Kepler figures out that, oh yeah, the sun really is at the center. Copernicus and all the rest. So I just think precision measurement, answering basic questions, and that's an unsolicited take from a proteomics technologist. So with that interjected into your day, I'm going to pivot over to Sean, and he's teed up, ready to go, talk about discovery neuroscience at Merck. Please, Sean, take it away.
Great. Neil, can you hear me? All
yes,
right, perfect.
Yes, very much. Yeah.
I was doing audio video check. Well, I appreciate the invitation to speak today about discovery neuroscience at Merck. And I listened to the panel discussion, and I think I'll touch upon some of the topics the panel was discussing and see there's some synergy between our thought process, which is always good. So first, a brief introduction to myself. So I'm a Discovery Neuroscience. I'm an executive director, and I had 3 departments focused on developing novel therapeutics for treating neurodegenerative diseases as well as psychiatric disorders. And my groups touch upon everything from target ID and validation through phase 2 clinical proof of concept. So a fairly broad range of the early discovery into preclinical into early clinical development. So introduction to Merck. I think a lot of people know, heard of the company Merck before. It's a traditional large pharmaceutical company headquartered in Kenilworth, New Jersey. There's about 74,000 employees worldwide. However, the main business focus of Merck are prescription medicines, vaccines, biological therapies, as well as animal health products. And Merck is still a very research-intensive organization with a focus on R&D, and our annual R&D budget is about $13.6 billion a year. So there's a heavy focus on research and development. Highlighting 3 sites on this slide: West Point, PA, which is outside of Philadelphia, Boston, and London, because these are the 3 sites where neuroscience research is conducted. So I just kind of wanted to introduce that to the group today. So before I dive into the specifics of how we conduct neuroscience research at Merck, I wanted to kind of introduce, the problem statement. I think the panel did a great job on discussing these issues earlier. Where basically, I think and I think most people can agree, there's a very high unmet medical need for CNS disorders, right? So these are diverse diseases that affect a large patient population. If you could read the graph on the right here, you could see of the top 10 therapeutic areas by global disability adjusted life years, neuroscience comes in second to cardiovascular disease, and it's been growing steadily from 2007 through 2017 and now through 2021. And the reason for this is really driven by the growing aging population, and also, also the ineffectiveness of current interventions, right? The therapies we have for CNS diseases aren't great, and there's dramatic room for improvement, in particular in areas for disease modification, but also there is still some room for symptomatic treatment as well. Traditionally, developing therapeutics for neuroscience has been one of the more challenging areas due to a number of reasons. For example, the restricted access of molecules into the brain, small molecules, large molecules, gene therapy challenges. In addition to that, it's very challenging to understand target engagement. You can't directly biopsy the tissue in the CNS. CSF measurements aren't ideal. And now with imaging techniques, these things have started to improve. And I think that's been highlighted, by the discussions you had earlier, where a greater understanding of the biology of these CNS diseases, as well as the improved biomarkers, are really increasing our POS of developing neuroscience therapeutics. I think we're kind of reaching a sweet spot where we kind of know who to target and when to target, and we're starting to see some level of success. So this is kind of highlighted. I won't talk about aducanumab, but I'll talk a little bit about another Alzheimer's trial. So Lilly recently showed some interesting data with their anti-amyloid therapy, donanemab. And really what they did is more of a precision medicine type experiment in Alzheimer's patients, where they are really able to target a specific disease stage and patient population where they had a particular burden of amyloid, but not tau. And they were able to at least show some disease modification effects of their antibody. And that's moving on to clinical development. But I do think our increased understanding of the staging of Alzheimer's disease, as well as the biomarkers available to clinicians that test these therapies, really enables us to develop new therapies to treat Alzheimer's disease. And we'll see what happens going forward with additional anti-tau therapies and additional therapies. Another area where we're starting to see some improvement is in the area of schizophrenia, where we have not necessarily better biomarkers, but a better understanding of the mechanism of disease. And we're really starting to see potentially some progress in treating the residual symptoms of schizophrenia by understanding this complex biology. And I think, that the novel therapeutics targeting NMDA receptor hypofunction are really kind of enabling us to move ahead in treating that disease. And BI had a nice finding recently with their GlyT1 inhibitor showing improvement, at least in some areas of cognition, in treating patients with schizophrenia. So As the panel kind of highlighted, I think there is a high unmet medical need. I still think there's several challenges with developing neuroscience therapeutics. However, I do think we're coming to a stage where we could really understand what patients we're enrolling in trials, and we can understand what mechanisms to target for these patients. And one last slide, I think this is actually translating to approvals by the FDA, obviously the one we just discussed from Biogen a few weeks ago. But I do think, if you look at 2020, neuroscience was only second to oncology for approvals in the FDA. So I do think we're starting to see some success in the area of neuroscience research. With that, I'll pivot a bit to kind of how we conduct neuroscience research at Merck. And over the years, we've kind of evolved our thinking. I think, several years ago, I started at Merck about 17 years ago, and we were more focused on using animal models to predict outcomes in the clinic. And I think through a lot of challenges with translation, we decided to kind of change our approach and really focus on using human biology to select the best drug targets. And how do we do that? I think it's a combination of really understanding the human genetics. Obviously, Myung just presented in the panel presentation, and he's one of my colleagues leading our genetic research at Merck. But partnering with groups like him are really starting to understand information from familial mutations or from genome-wide association studies, combining that with an understanding of pathophysiology of tissue. So really understanding, looking at human tissue, what organs are impacted or what cells are impacted, what type of proteins are altered. We could really come up with, intriguing hypotheses of what's going on in these diseases and really make an educated hypothesis of what to target, right? So if we understand where mutations are and we understand what the proteins are doing, we have a better idea of what proteins to target. And this leads us to develop these mechanism-based hypotheses, things like looking at neuroinflammation, mitochondrial dysfunction, lysosomal impairment. And we could select therapeutic targets and really build research operating plans based in human biology, right? So taking patient-derived iPSC cells or having human cellular models that recapitulate the disease and really build the right pharmacology to address those models. And I do think in vivo models or animal models have a role, but it's more for understanding, PK, distribution, target engagement, pharmacodynamic effects. So we could really target the processes we're interested in and understand how those doses and exposures relate to biomarker changes, right? So once we understand our targets, we have an idea of the biology, we really want to make sure everything's in place to translate that into the clinic, right? And I think the key areas for that is really understanding dose, exposure, target engagement, and having the right tools to clearly demonstrate in early phase 1 and 1A and 1B studies that you're not only getting adequate exposure, but engaging the target and having a biological effect that's adequate to test a mechanism in a disease of interest. So I think once we have all these things in place, we're in a better position to truly know if a mechanism works or doesn't work, right? The worst case scenario you could be in is if you take a target into the clinic and you're unsure if you engage that target with a negative outcome, right? So at least if it's negative, you fully engage the target, you got the right exposure, you hit the right biomarkers, at least you can walk away from a particular target if you adequately tested that. So I think we're in a good position now in neuroscience to do that. And that's kind of how we're proceeding at Merck. So as we, really investigated human cells and genetics, we really, came to an understanding that Alzheimer's is not purely distinct from Parkinson's. It's not purely distinct from ALS. It's not purely distinct from Huntington's disease. There's there's a lot of similarities between these diseases, and really there's common mechanisms that affect the progression of these diseases, right? So if you look at in Alzheimer's postmortem tissue, you do see altered proteostasis. You see amyloid protein, you see tau protein altered. Same thing in Parkinson's synuclein, right? So I think there's common mechanisms that really underlie a lot of these diseases. So if you look at dysregulated proteostasis, lysosomal dysfunction, mitochondrial dysfunction, and immune dysregulation, these are common themes you see across multiple diseases. So based on these, common findings, what Merck decided to do was not necessarily have a group doing Alzheimer's research or a group doing Parkinson's research. we're more focused on these underlying, what we call buckets of biology. So we have groups focused on really understanding these underlying processes that may be applied to multiple distinct diseases. And that's kind of what the organization is at Merck, you see on the screen. So in the different locations, for example, outside of Philadelphia at West Point, we're focused on understanding things such as altered proteostasis, lipid regulation, lysosomal biology. In addition to that, we have a lot of work going on for symptomatic treatments, really understanding the basis of synaptic biology, ion channel functioning, cell signaling, et cetera. In Boston, the teams are more focused on neuroimmunology, looking more at myeloid cell biology and neuroinflammation. And in London, the new groups are looking at mitophagy and oxidative stress. So I think really as we learn more about these processes and we're able to understand potentially how different targets may impact these processes, we'll select which diseases may be best to test these in the clinic. I think one of the panelists mentioned which one goes first. I think that's a very interesting question we're trying to grapple with. Is it a combination of finding the right patients, having the right biomarkers, having a clear pathology? I think that's things we could discuss moving ahead. So to kind of close in this section, I want to give an example of an area that we find quite exciting. It's an area of autophagy. I think a number of genetic studies as well as pathological data really link deficits in autophagy to neurodegenerative disease, right? So I think autophagy, if you don't know what it is, it's a process for removing toxic proteins and damaged organelles to maintain cellular homeostasis. If you think about autophagy, you should think about lysosomal functioning, right? So it basically uses proteolytic degradation of cytosolic components to clear these toxic proteins. Also, it's interesting that this function of autophagy is really a defining hallmark of most neurodegenerative diseases characterized by accumulation of toxic proteins. So we found issues with autophagy in Alzheimer's disease, Parkinson's, Huntington's disease, et cetera. And evidence has also accumulated recently to support that enhancing lysosomal function and autophagy actually may protect against neurodegeneration. And one protein of interest, I think, or transcription factor of interest is TFEB. People call this a master regulator of autophagy, but this has emerged as an important target to modulate autophagy and maintain cellular homeostasis. And one particular pharmacological target that may impact TFEB and autophagy is TRPM01. And this is really, one of, we think, the more tractable drug targets for TFEB modulation. And this is something we've recently been pursuing following an acquisition of a small company called Calporta. And we think there's a lot of promise in this potential mechanism in regulating autophagy, and whether we go into a larger disease or a small disease first, I think that remains to be seen. But, in kind of closing, I do think if we take a step back and look at how we perceive a drug development, necessarily not from a disease perspective, but from a mechanism perspective, we actually may have more luck in really defining key mechanisms that may impact multiple diseases and have multiple shots on goal to impact, this high unmet medical need. And with that, I'll take a break and thank people for their attention, and I get to stop and take some questions.
Okay, Sean, thank you for that. And the floor is open for a question or two.
May I ask a question?
Please jump right in, honey.
This was excellent, and I was very happy to hear that Merck is actually focusing on mechanisms rather than disease names. And I think, when diseases were characterized, this was like 100 years ago, right? Dr. Parkinson, Dr. Alzheimer's, it was all clinical. But at the time, we didn't have proteomics, we didn't have genetics, we didn't have the information that we have. And finding the common mechanisms are very important, and I applaud you for focusing on autophagy. So that's a very important one for ALS as well.
Thanks. Appreciate the feedback.
Over at Merck, we had someone just be interested in, you and Myung run pretty big machines. How has the pandemic impacted your operation? Has it?
Yeah, It's interesting because what you expect would happen really didn't. So we were really concerned early on with clinical trials and understanding if patients would come to the clinic and understand if we could continue the trial. And in fact, those kind of proceeded quite well. We had a focus on key trials and they were supported as far as getting, the right supplies to the sites and getting the patients, monitored. I think where the surprises came up is more in the day-to-day functioning of the preclinical space, getting, lab supplies and plastics and cell culture wells and things like that actually have been the bigger issue.
Yeah.
And other than kind of, and I'm sure a lot of people feel that pain across the board, really understanding, reaching far and wide to get the right materials to run your preclinical models, right? Even getting, preclinical species for experiments has been a bit challenging too. So I do think it's kind of been the supply at the back end more than the front end of the patients.
All right, thanks for that Merck perspective on that. Well, one more here from Len. Many thanks for your contribution. And what's the time frame to clinic for the TRPML1 drug? I fear I might miss the revolution.
Yeah, so obviously, we don't give minute-to-minute updates in this, but I would say I think from the Cal Porter perspective that we are kind of in the preclinical stage, really going through more discovery. So I think we're a few years out from our initial clinical trial and understanding what this mechanism has. So I think we have a few more years before our clinical studies.
Okay, Sean, thanks so much for that. And I love your mental frequency. It matched my own. I really enjoyed that a lot. And so thanks to Sean, and we're going to pivot over to Casper for his installment. Here we are. He's VP, Head of Neuroscience Research at Genentech, and we're all waiting for your contribution, Casper. So I give you, without any further ado, Casper Hugenrod.
Thank you so much. I'm just doing an audio check here. Can you hear me?
Absolutely, loud and clear.
Great. So let's see if I can forward the slides. Perfect. So good morning, everyone, or good afternoon. Happy to give you an overview of what Genentech and Roche are doing in the neuroscience space. Maybe just one sentence about myself. I'm heading up now neuroscience here at Genentech for the last 4 years. Before that, I was actually a professor in molecular neuroscience and was leading a department for the last 10 years before going to Genentech, which was called the Department of Cell Biology, Neurobiology, and Biophysics in Utrecht University. So what I'm going to do today is going to walk you through what Roche is doing, what Genentech is doing in target neurodegenerative diseases. And we're going to talk you from a commercial landscape to late stage to early development to basically discovery research. So taking actually a reverse look at it. So let me start with why we are in neuroscience and just show you some facts and figures behind what drives us in neuroscience in the portfolio. And I don't think I have to explain it again because Sean did an amazing job and the panel discussion was there. I mean, neuroscience remains an area of a great unmet medical need and poses one of the greatest healthcare challenges of our time. And just to give you again some of the numbers, there's more than 600 neurological diseases, 700 million cases of disorders reported globally each year, and around $800 billion in the US alone on healthcare costs. And this is another 800 billion in Europe. So what are we doing in Roche and Genentech? What's our commitment? We have currently 14 medicines in neuroscience in the clinic, around 115 clinical trials globally. We already have 60 years of neuroscience experience. I will come back to that in a second. And currently they're working more than 4,000 scientists dedicated and focused on these neuroscience and neurological diseases. So just to give you our history in neuroscience, in Roche, it already started in 1960, where we actually discovered Valium for anxiety disorders. In 1973, we got pretty good results in Parkinson's disease with L-DOPA and a combination with the carboxylase inhibitor benzoradazide. In 1996, we had a molecule and which was a closed dissolving medicine, which is called Activase. It was approved for acute ischemic stroke. And a little bit more recently, we had a medicine which is called Ocrevus, approved for MS, both for relapsing and for primary progressive MS, which targets CD20 on positive B cells. And last year was really a good year. We had two molecules approved. One is Evrysdi, which is actually called risdiplam before, which is a very interesting MOA. It's a splice modulator for spinal muscular atrophy. So it's an SMN2 splice modulator. And another molecule which inhibits IL-6 receptor activity for neuromyelitis optica spectrum disorders is called Enspryng. So just to give you a little bit of the highlights we had over the years in neuroscience in Roche Genentech. This is the current portfolio. I'm not going to walk you through the current portfolio. This is the clinical portfolio. You know, we have focus on neuroimmunology, neurodegeneration. I will come back to the neurodegeneration part because it's the topic of today. Neuromuscular diseases, neurodevelopmental diseases, psychiatric diseases, schizophrenia, autism, etc., etc. Again, I'm not going to walk you through all of them. The molecules are listed there. Many of them, maybe that's good to highlight, are partnered with other companies, and some of the molecules we developed internally, and they're either in phase 1, 2, or 3. So as I said, this is the clinical portfolio, but I would like to tell you a little bit more about how we think about the next generation of medicine and the next generation of drugs at Genentech. So I'm going to move now more to the discovery research and the early clinical development and discovery part of what we're doing at Genentech. And the mission is really threefold. One is to focus very heavily on preventing and slowing down the neurodegenerative diseases by early treatment of key pathogenic mechanisms. And I cannot emphasize more the word key pathogenic, because I think that is really where we want to go if you want to slow down and even prevent these diseases. We also have a big effort in repairing and regeneration the nervous system. Just one example is remyelination. So thinking about MS, we have an immune regulator now with Ocrevus, a CD20, and the next thing to do in MS is really to help these patients with remyelinating and bringing back functions. And we're having big efforts in that. It is a kind of a cellular therapy approach. You can think about different cellular therapies, thinking about iPS cells and other therapies where you can actually repair and restore function. And we're very, very interested in that. The third bullet is also a big pillar for us, which is severe chronic pain, such as neuropathies and diabetic neuropathies. And we're following here the human genetics. We have a big investment in understanding the human genetics behind pain and understanding basic neurological mechanisms. For example, the mechanisms, the neuronal circuitry in the spinal cord around pain. I will not focus on that, but I'm happy to answer questions about our efforts in pain. So just on neurodegeneration, what are the diseases? Again, it's very similar to what we're doing in the clinic. I would say, you know, Alzheimer's disease is a big one. MS, I already told you about the remyelination efforts we're having. Parkinson's disease, glaucoma is very interesting for us as well, because we really think you also want to protect neurons from dying in glaucoma, and that is a big effort. ALS, we're very interested in, FTD and tauopathies. And these last three diseases are always linked, I would say, to the bigger indications. And it comes back to the discussion, if we find a drug and a mechanism of action, what is the disease and what is the indication we want to go into? Very similar to what is discussed just previously. But very similar to what Sean was saying, I am not that focused on these diseases. I'm really more focused on the biology and the mechanisms behind these diseases. And at Genentech, we bucket them in 4 buckets, I would say, 4 teams of biology. And I will walk through all 4 of them just to give you a little bit of a flavor what we think is going on. And you will see, you know, all of them probably contribute at some point to the neurodegenerative diseases and the indications I just mentioned. So the first one is focusing on aggregation and cellular homeostasis. Basically, we have very basic, almost academic questions. Why are these proteins actually forming aggregate? And we're talking about TDP-43, SOT1, A beta, the amyloid plaques, tau aggregates, C9orf. So why are these aggregates formed, and why are they formed in a cell-specific manner? Why are they not cleared? And in some cases, even there's evidence of spreading of these toxic proteins, and why are they spread from cell to cell? So these are basic cellular mechanisms, and we try to find inhibitors and in each of these pathways. The second bucket, the second theme is neuroimmune communication. And I don't think I have to convince you that there's a lot of human genetic data nowadays in Alzheimer's disease, that many of the human genetic hits actually are expressed, and the risk factors are expressed in microglia cells. So we're very interested what the role of microglia cells or glia cell biology is in the brain. And we're having topics around, is it actually beneficial, these microglia cells? You can imagine that if you activate them, that they clear up the plaques. But on the other hand, there's a lot of evidence that overactivation of these microglia cells can be actually harmful in Alzheimer's disease. So we're thinking about very clearly from a human genetic perspective, but also understanding the basic biology behind microglia cells and other glia cells in the brain. The third theme we have is around modulation of neural circuitry. This also comes back a little bit what was discussed about schizophrenia and other more psychiatric diseases, but we believe that an imbalance in excitatory over inhibitory cell signals are also very important in the progression of Alzheimer's disease. So we're having also very significant efforts in that area. And the fourth theme I already kind of discussed with you is about repair and regeneration. What can we do with cellular therapies? What can we do about, for example, again, remyelination? And can we also protect neurons from dying in neurodegenerative disease? What seems to be, one of the big themes we're focusing on. Can we protect just neurons from dying wherever the toxicity comes from? So this is just to give you a flavor of the mechanisms and the hypothesis we have and the kind of drugs we want to develop in the coming years. Just to give you one highlight, and I just want to mention one of the programs, or actually a couple of programs that we're very focused on, is the protein tau. And this is also not a new protein, obviously, in neurodegenerative diseases, but there's such a clear evidence that the tau pathology and the distribution of tau in the brain, but also the density of these plaques and tangles are so significantly associated with the cognitive and clinical disease severity in Alzheimer's disease. And just to give you like two examples of a correlation between the cognitive score and the number of tangles found in the brain, and our GTP1 tau PET tracer, which is a genetic PET tracer, which correlates very nicely with the progression of Alzheimer's disease, and which could be used as a potential surrogate biomarker moving forward. So we're very excited about that. And we have two large molecule efforts around tau. One is bepranemab, which is a mid-domain targeting tau, a large molecule antibody. And we're starting the phase 2 basically right now, which is partnered with UCB Pharma. And our second approach is the semenorumab antibody, which is an N-terminal antibody against tau. We already have read out the prodromal to mild AD in 2020, and currently we're working on the moderate AD population. And basically the therapeutic hypothesis is here that tau species, misfolded forms or fragments, are basically spreading through the brain and work as potential seeds for new tau aggregates to form. So here's a cartoon where we see in red the antibodies basically grabbing the extracellular tau and slowing down the progression of the spread of this tau species. So again, we have two approaches here. And in the next two slides, I'll just show you a little bit of the in vivo preclinical evidence that we have. Here is the murine semenorumab antibody, which is the N-terminal tau, which shows that in animal models, we have a dose-respondent response if we block the spreading of tau in these animal models going forward when they age. This is published in two papers recently as well. And very similar data, I would say, with our murine bepranemab antibody, which also slows down the progression very nicely in these animal models. both in this case in the IPSI and the contralateral where the tau was injected. Coming back to the discussion about new modalities, we're very, very interested in it. I think it makes a lot of sense if we have a good target and we cannot drug it with a small or a large molecule, that we find other ways of doing that. We're very interested in an ASO and antisense technologies. I already told you about the splice modulators. We're very interested in that to change the RNA splicing or just go with small molecules after RNA interactors. Cellular therapies I already mentioned. And together with Spark, we're very interested in developing new gene therapies. And again, in the panel discussions, it was already mentioned what the challenges are there. But I think it is good to think about these new kind of approaches, because if such a platform is safe and it's going to work, it will open up a lot of possibilities. So again, one of the challenges is obviously brain delivery. This is both with ASOs, siRNAs, intrathecal delivery, other ways of delivering it in the brain, and gene therapy. It would be obviously fantastic if you have a systemic delivery of gene therapy that directly goes to the brain, to the right cell type, delivers the cargo. We're not there yet, but I think this is something we really want to work towards. So just thinking about how can we find new targets in that space, and in these 4 therapeutic, I would say, themes, the biological themes I was talking about. And I have a very similar slide, actually, what Sean showed, is that we're really heavily relying on human genetics. We have a lot of patient data. We have run these Alzheimer's trials. We sequence the patients. We have a lot of human genetic data. And of course, we're leveraging all kinds of external databases. We're having strong computational approaches just to understand what are the key pathways in Alzheimer's disease, Parkinson's disease. We do a lot of molecular phenotyping, a huge amount of gene expression analysis. You're probably aware that Aviv Regev just joined Genentech as head of GRED, and there's a lot of efforts now to make these beautiful atlases again, but now for disease states in human brain and focus on that. So I'm very, very happy with that. And maybe the last thing to mention is we're really doubling down on functional genomic screens. So once we have a human genetic hit, to understand, in what pathway is it exactly, and I want to do that in the right cell type, obviously. I mean, it will create new hypotheses about pathways, but sometimes in a pathway when we have a clear human genetic hits and there's some challenge about druggability, we maybe find within the pathway specific targets that we can target and is more druggable in that way. So doing this approach will not only find new targets, but also find new biomarkers, obviously. And I already mentioned that we want to do it in the right cell type. Was mentioned earlier that mouse models, I would say, have their limitations. And we're really doubling down now on going to iPS cells, where you can nicely genetically manipulate, of course, the risk factors, for example, growing organoids, and even trying to do not only functional genomic screens, but even small molecule screens in these, I would say, more challenging but very relevant human cell types. And again, I cannot emphasize enough, and I'm just going to repeat again what the panel was already discussed, how important it is to have the right biomarker. Either, just the example of GTP1, which is PET tracer, but also genetic biomarkers, fluid biomarkers, or any other biomarkers that can read out the progression of the disease is going to be so important moving forward in any of the neurodegenerative diseases. And in the last 2 slides, I just want to highlight one of the big efforts we are currently having, and this was announced at the beginning of the year, where we wanted to expand the Roche Genentech neuroscience drug discovery efforts. And we're doing that by collaborating with leading academics on, I would say, very early, high risk basic research. And this was, we decided to do a collaboration with the Weil Neuro Hub, which is actually a joint research collaboration, what was already existing between UCSF, UC Berkeley, and the University of Washington. And this is a long-term research collaboration commitment that Genentech and Roche did, which is a 53 million total funding for the coming years. And I'm really, really excited about this because it's all project-based, and it's fantastic, of course, to work with all these great scientists. And this is the last slide, just to give you a little bit more detail about the breadth of this collaboration. UCSF has more than 500 neuroscience and psychiatric faculties. Berkeley, more than 120 neuroscience researchers, which are just amazing. And combining that with our strength at Roche to develop drugs, I think will really emphasize, the discovery aspect in neuroscience, potential of translatability, but also be able to do clinical trials together with UCSF. One of the examples we had was actually developing together Ocrevus, and UCSF and Genentech has been very fruitful in that collaboration. And I think this would be an engine, of getting really great new discoveries in neuroscience, not only on targets, not only on translatable biomarkers, but also even on thinking about digital tools or other devices that we can help patients going forward. And with that, I just want to thank everyone for listening to the talk, and I'm happy to take any questions.
Jasper, thank you so much for that installment, and a lot of exciting things happening out there, South San Fran. The floor is open for questions. I think there's one about the comment on tau. Could you comment on that, like your, Genentech's engagement with tau, and then recent developments about that target, either as a marker or the types of proteins that are recognized by your antibodies?
Yeah, so we read out the seminurumab, which is the N-terminal tau, which was in prodromal to mild patients. And I think it was mentioned also in the Q&A that the Biogen data actually was, like our data, was unfortunately not positive. However, we learned still a lot from the trial. For example, just to give you again, about the GTP1, we basically saw that it correlates very well with the progression of the disease. So we're very happy that, again, that repeats. As a therapeutic, we need to see how the molecule behaves. That's why we are also thinking about the mid-domain antibody and with the UCB collaboration. And also thinking about, and we're running the trials obviously in different populations of people with Alzheimer's disease, for example, the moderate population. So we'll just need to see and how that plays out in the clinic. But I think it's still very exciting to work on tau, to work on the spreading hypothesis, but also to come back, I think, to the new modalities. And was mentioned before, it's also about the small molecule approaches that we can do on tau, or other approaches, more genetic approaches. And I think tau is a good target, and we should really double down on that target and understand it really, really clearly.
Yeah, I completely agree with that. There's 6 isoforms, 65 known phosphorylation sites. The proteoforms of that are completely unknown. And this is what the kind of things that I'm very passionate about. But I'm going to set that aside. It's not about me. We're going to move on to the Q&A. And one of it's a sprawling type of interactions and collaborations. It is kind of moving toward a wider lens and wider network of collaborations. Bordering on pre-competitive. One person wants to know: did you reach out to the Allen Institute? Are they on your radar at all? The Allen Brain Institute.
Yeah, I mean, we're very interested in collaborating with many, I would say, academic institutes and other institutes in the area. And this is really an active, I would say the NeuroHub and the Weil Institute is probably just the beginning, but this is really something that we're very passionate about, and working more with academic labs and going forward. And obviously, the Allen Brain Institute is just one example, and it is great to make connections there. But there are several others on our radar that we could collaborate with. Absolutely.
Right. I've got maybe one more here. Let's see. Are there any non-tau A-beta complementary approaches? For example, glymphatic system approaches that you've thought about?
Yeah, no, this is a great question. We're really interested in glymphatic systems and other ways of doing it. I would say currently, I think after the A-beta and the taus, the next is probably microglia cell targets that are coming into the clinic. So the microglia cell modulations, I think that's probably the next wave of new, I would say, MOAs that we need to be tested, and for good reasons. Yeah, definitely. This would be the fourth generation probably.
Yeah, and I agree with that. I think a lot of interest in TREM2, for example, is an area of research for a lot of companies related to neuroimmunology. So I think I totally agree with that.
Thanks, Sean.
Interesting. There's a new way of thinking. Yeah, that's great. Maybe we'll give Hande we'll end where we started, and that was with Hande here. She's asking an edgy question about FDA. So when you develop a compound based on mechanism, how would you plan to apply for FDA approval if the mechanism is shared among multiple diseases? Would it be possible to include different patient populations in the same clinical trial? That's out of the box kind of thinking. Like with your genetics, your human genetics that you have, could you bin them in ways that were different than disease names?
I think it's out of the box thinking, but I don't think it's completely crazy. I like it. No, I like it. I like the idea. If you can stratify patients in a different way, I think it makes a lot of sense. It's not conventional, but from a scientific point of view, it makes a lot of sense. Yeah.
An example of that is Acadia Pharmaceuticals recently had a trial from the compound pimavanserin looking more at behavior-related disturbances in dementia, and they're looking across a broad panel of dementias, right? So that's one example of common symptoms across multiple diseases, and they have to do a little bit more work there. But I do think, potentially looking at, combining multiple endpoints in one trial might be hard, but maybe doing a couple of trials to support a common mechanism may be reasonable for a broad approval.
And this has happened in the cancer oncology space, has it not? Where different driving mutations across different, in lung or pancreatic, et cetera, like the KRAS cysteine mutant, et cetera. Okay, well, that was spectacular. Sean, Casper, thank you so much for contributing today and elevating our game. We're going to move to the last session, and that's chaired by Jay Olson from Oppenheimer. And Jay, I think it's all about you. Take us home to the top of the hour, and we'll be in great shape.
Thank you, Neil. On behalf of Oppenheimer, we really appreciate the collaboration, and it's been a pleasure working with you, Sheila, Lisa, and the amazing faculty at Northwestern to put together this exciting event, along with Lauren and our partners at BioCentury. I'm Jay Olson. I'm a biotech research analyst at Oppenheimer. I want to welcome our distinguished panelists and thank them for joining us today. I'm going to ask each of you to briefly introduce yourselves, and we'll go in alphabetical order. So Jamil, if you could please get us started, that would be great.
Sure, and I'll just do a quick mic check. Can you hear me?
Yes, we can.
Great. So Jamil Beg, good morning, everybody. Good afternoon as well, and thanks for the invitation to participate. I'm a partner at 5AM Ventures, and just very briefly, 15, 16 years in the industry. I've operated 4 venture-backed startups, 3 of which were neuroscience companies in business development, commercial leadership, and CEO roles, raising several $100 million across private and public financings. And most important to me, worked on 2 therapeutics approved by the FDA across disease areas. Very briefly, 5AM Ventures, early stage life science venture fund, seed Series A, first institutional investor is what we aim to be. About 20 years of investing, $2 billion under management. We're investing in North America, Western Europe. We're always leading or co-leading financings. The vast majority of our investments are in novel therapeutic platforms, therapeutic area agnostic, modality agnostic, and have invested heavily in neuroscience over the last few years. Total of about 90 companies we've invested and 15 approved therapeutics from those portfolio companies over that 20-year history.
Thank you, Jamil. Margarita?
Hello, everyone. Oops, sorry. Just gonna do a mic check.
That's better.
Can you hear me?
Yes.
Go ahead. Sorry. Can you hear me? Margarita Chavez is AbbVie Ventures, a managing director. AbbVie Ventures is a strategic venture fund with AbbVie as its sole LP. We've been around for about a decade now. Have about 2 dozen active companies currently, and we have invested about twice that much in the past 10 years. I guess kind of young compared to 5 a.m. Very selective and strategic in the way we invest. And what that means is, we invest in companies with science and technology that has potential to be transformational in areas that are focused to AbbVie. It's oncology, neurodegenerative disease, immunology, and ophthalmology. And for me, I've been, like I said, I've been with the venture group since its inception. I'm proud to be one of its founding members. Was in BD before that, did for about 5 years. Did some interesting deals for AbbVie, including the one that brought Elagolix and the Lupron franchise into the company. So excited to be here.
Thank you, Margarita. Michael? Michael, you might be muted.
You hear me? Now you can hear me.
Yeah.
Good afternoon, everyone. Thanks for having us on the panel today. Jay, good to see you. I think this is our 3rd year in a row doing this. I guess 2nd year in a row doing it virtually. So I earned for going back to Northwestern and staring at the beautiful Lake Michigan. So hopefully that'll happen next year. I'm Michael Margolis, Senior Managing Director. I head up the Life Science Investment Banking Group at Oppenheimer. I've been here almost 5 years, almost 25 years in life sciences, started life as a scientist, worked in big pharma, both Lilly and Novartis, worked in Basel at Novartis and Global BD now in new product roles. And I've been a banker since 2006. And at Oppenheimer, we focus on emerging growth companies, very active middle market firm, really focused on IPOs, secondaries, really the full spectrum of investment banking capabilities for our clients. Certainly, very excited to be part of this discussion. I think from a neuroscience standpoint, a lot of excitement in our space. Great to see a recent approval by Biogen, and certainly a lot of excitement in the pipelines, including Roche and others at this point.
Thank you, Michael. Jingwen?
Can you hear me?
Yes.
Great. First of all, thanks, Jay, for organizing and to Northwestern. I'm Jingwen Wang. I manage healthcare investments for Verition Fund Management. We're a multi-strategy investment firm with $4 billion under management. And I invest in both public and private biotech and pharma companies. I am a neuroscientist by training. I got my PhD in neuropsychopharmacology at Columbia and initially wanted to do R&D in biotech, but in grad school became fascinated by being at the intersection of science, medicine, and the market. So after grad school, I spent 4 years at McKinsey in the healthcare practice, working with biotech and pharma companies on drug development and commercialization. And about 7-8 years ago, moved onto the investment side. And naturally, because of my background and having worked with quite a few CNS companies in the past, I end up looking at a lot of investment opportunities within neuroscience.
Thank you, Jingwen, and thank all of you for joining us here today. We're going into the home stretch, and this is sort of the lightning round. And I just want to provide a short background before we begin our discussion. So I'm going to quickly go through these slides, starting with these first couple of slides showing that biotech is slightly underperforming the broader market year to date with a rotation out of mid-cap or small-cap biotech, despite record numbers of FDA approvals in recent years. As shown on this slide, FDA approvals are an important catalyst for biotech stocks. For example, as we've spoken about today, FDA's approval of aducanumab. And venture capital is an important source of funding for emerging biotech companies, as we will be discussing. And sources of funding include the classic VC funds like Sophie Nova, and also corporate funds like AbbVie Ventures. And within VC funding, neuroscience ranks among the top 3 areas of VC investing. Other important events in biotech investing include IPOs and M&A activity. These are key drivers and indicators of biotech investment performance. And although biotech deals were down slightly during the pandemic, it started to pick up again recently. And biotech IPO performance has been strong. And surprisingly, 2021 is already looking like a record year for IPOs, even though we're only halfway through the year. So with that, let's jump into our questions. And as Neil and others have pointed out, the timing of today's event with our focus on neuroscience is very appropriate. And there are a lot of implications from Biogen to other companies working in neurodegeneration. So our first question is, how does the FDA approval of aducanumab impact your view of financing or investing in CNS therapeutics? And Margarita, if you could get us started, that would be great.
Sure. Yeah. I mean, it's, I think, exciting for the area that there's a new drug approved by the FDA for Alzheimer's. Is definitely an area of high unmet need, an area of interest to us. Doesn't really change much with the way we invest. Again, we invest in companies that are working on science that's novel and potentially transformational for patients with a disease. And so we have not really, and will not probably be focusing in that particular pathway, but we'll continue to look for novel modalities and novel pathways in autophagy and as was discussed earlier by Casper and tau and like I said, other modalities. But I am hoping that with this approval that emboldens other innovators, including researchers and investors, to do more and expend more resources in the area.
Great, thank you, Margarita. Jamil?
Yeah, similarly, our investment strategy hasn't fundamentally changed. I think it's really encouraging to see from the FDA kind of flexibility around endpoints and the potential to see a path to accelerated approvals. And I think we can spend the entire day, if we wanted to, talking about aducanumab. So I'll save that debate for another day. But I do think it will drive more broad interest in private financings and public financings, and give neuroscience companies more options going forward to take programs further into clinical proof of concept studies, clinical registration studies as a standalone, and not necessarily be dependent upon partnering with larger biopharmas. And so I think that's net a good thing.
Great, thank you, Jamil. Michael, would you like to add anything?
Sure, I bring it from a different perspective, more from the public markets. Look, I think Biogen's approval, I think is, it's very encouraging. It was the first drug approved for Alzheimer's since 2003, so 18 years. I think it also shows you the FDA is very accommodating. I think a lot of folks were surprised, especially on the buy side. I think, there are definitely people that are out there who are very cynical, but it shows you the hurdle rate's quite low for an Alzheimer's drug. So I think for new drugs coming down the pike, I think, it's certainly, it's encouraging. The worry that I have is that, there is some kind of fallout from this approval. It's not a very clean drug. Are there going to be any kind of adverse events that I think put a black eye on the FDA down the road, which would make it more difficult for, future drug approvals, just from a safety hurdle? What's interesting is you look at a halo effect on the public side, you see a lot of early stage companies. And Jay, you and I talked about a couple of these yesterday. They've had a tremendous increase in valuation. Some of them are upwards of $3 billion, almost $2 billion, $1 billion. So these are very, very early stage assets that probably don't have the scientific rigor that other companies, I guess, go through. So that's the risk as well. Being as an investor, you got to be careful. But I think it's very exciting. I started my career at Lilly in neuroscience, with depression and certainly looking at atypical antipsychotics. So I think for me to see this momentum in the space, especially looking at neurodegenerative disorders, I think it's super important. But I'm also a little cautious about reading into this too much. I think time will tell.
Great, thank you for that perspective. Many of us wonder if biotech investors have traditionally shied away from neuroscience due to the risks involved, and therefore potentially create underappreciated investing opportunities. So with that, maybe if you could talk about what is an investable thesis in neuroscience. And Jing-Wen, would you like to start?
Yes, definitely. And just speaking of, I don't think it's just investors shying away from CNS, but we've also seen quite a few big pharma and biotech moving away from CNS in the last 5 to 10 years. And I mean, I understand why, because it is very difficult to do rationalized drug development in neuroscience, because a lot is unknown about the disease itself. The pathophysiology of the disease is often very complex and heterogeneous. And then you have endpoints that are sometimes highly subjective and therefore creating potential noise and bias. So with all of that said, when it comes to investable or investment thesis, I think a few areas that I look at, the relatively easier one, and I think some of the earlier panels spoke to this, is if you have a precision medicine approach that directly targets the causality of the disease. So for example, some of the SMN targeted therapies for SMA. But unfortunately, most of CNS diseases don't fall into this category. So then the other two things I look at is, do you see, especially in smaller data sets and earlier stage trials, do you see very clear signs or signals of efficacy that cannot be explained by either placebo effect or bias? And again, the SMN targeted gene therapies or ASOs are a great example. But also some of the rapid acting antidepressants like ketamine, where when you look at both magnitude of effect and rapid onset of action, it really can't be explained by anything other than the fact that this is an active drug for this disease. And then the third category that I look at is more difficult, is if you're consistently seeing weak signs of efficacy either across different endpoints or different drugs in the same class. And I think beta amyloid is actually a good example of this, where you've seen across multiple different antibodies, either in certain trials or in certain patient population, you are consistently seeing a very weak efficacy signal. But then the key then is, how do you increase the efficacy signal by targeting a different area of the target or a specific patient population. And so that's normally how I approach it.
Great, thank you, Jingwen. Would you like to add anything, Jamil?
Yeah, I would just say I do think investors shy away from CNS due to the risks, but I think relative to a decade ago, there's significantly more interest in neuroscience broadly. I remember being at a Series A company trying to raise the Series B and not getting a lot of love in terms of first meetings. And so I think today it's very encouraging to see how much interest there is from an investment perspective. In terms of an investable thesis, I think there have been some great advances over the last decade in terms of the tools for R&D in neuroscience. And we think about genetic validation of targets or using human tissue-based samples, whether they're iPSC neurons or other tissue types, that can really help represent the human disease more so than artificial transgenic mouse models, for instance. I think everybody appreciates the challenges of in vivo models in neuropsychiatry and even neurodegeneration. And so having that fundamental support with human tissue is really important. Then thinking about what's a tractable translational plan to get to that rapid proof of concept in a human population, whether it's a healthy volunteer to show target engagement or brain penetration, whatever it may be, or even in a phase 1B where you see movement in a biomarker that gives you permission to believe that running the longer, more expensive, riskier clinical endpoint trial is going to be the right risk reward there.
Great, thank you, Jamil. Margarita?
Yeah, I would echo what's been said. You know, in neurodegeneration, at least, we've always been consistently interested in and invested in novel therapies and companies working novel therapies towards a disease. It is about having, identifying a genetic driver, a mechanistic approach towards the disease. And, I think a platform, an expertise in a particular pathway or a particular modality, I think as an investment thesis, to me is really very important.
having clinical validation, I think, makes it a lot easier. It's really hard to find in neuro. So I do think that if you have, as Jamil mentioned, identified at least these novel targets from either patient tissues, patient samples, or other types of, I guess, databases in proteomics or genomic databases. I think that's helpful, but you always need the biology, of course, because there are a couple of companies out there that are working on computational technologies to identify novel targets. I think it's still important really to generate the biology and develop a biologic hypothesis around what you're doing, especially in neuro. Neuropsych has been very challenging for us. AbbVie, our sole LP, obviously was not in neuropsych until we did the acquisition of Allergan. We have on-market products. We have a couple that are in the clinic. We are not investing currently, but I think the more comfortable we become with the genetic drivers of disease, which is likely going to be multiple, and as mentioned earlier, really probably going to require polypharm at some point. I think that might be a little bit more investable in the future.
Great, thank you. That's super helpful. And for the next question, I'd like to ask each of you to comment on new areas that you are most excited about in CNS therapeutics. And Jamil, would you like to go first?
Sure thing. And so hopefully I won't echo too much what the industry speakers, the R&D leaders said, but I think advanced modalities are a really exciting space. Gene therapy, cell therapy, genetic medicines broadly. I think many of you probably saw the Blue Rock just initiated a cell therapy a clinical trial. AbbVie just announced a collaboration in gene therapy, CNS gene therapy. I think the blood-brain barrier is still a big barrier, frankly, to large molecules. And thinking about vectorized monoclonal antibodies for really testing some of these hypotheses that you could argue haven't been fully tested yet in humans. So those are a couple from a modality perspective. In terms of area biologies, autophagy, hugely interesting, neuroinflammation, microglia are a particularly interesting area. I think they've been called DAMs, disease-associated microglia, and are they good or are they bad? Who knows? We're still figuring it out. Depends on the disease, depends on the state. And I think that will be continue to be an emerging field over the next several years. And then things like synaptic resilience and in the regulatory genome, as we call it, splice modifiers. And as we better understand disease pathophysiology, we'll have the tools in hand to then affect that. I'd also be remiss if I didn't mention a slightly side topic, prescription digital therapeutics. So this is a whole other modality that is emerging right now. And some may have seen that Pear Therapeutics just announced their SPAC this morning. And so they'll become a public company over the next few months. But really, they pioneered a field of applying the rigor of clinical drug development and the regulatory approval process to software to treat human diseases. And they have multiple approved products across neuropsychiatric diseases today. And I think that's going to be a field to continue to watch. There are now multiple players in there, and it's really a new subsector within healthcare.
Great, thank you, Jamil. Jingwen, would you like to comment?
Yeah, again, not to echo too much what's been said already. With new modalities, I think my thinking is I want to see new modalities that enables target engagement in ways that traditional approaches can't address. So take tau, for example, we've seen a few failures in tau. I think one of the key questions is, do you need the different binding region for the tau antibodies, or do you need to target intracellular tau? And there are a few novel approaches using new modalities that can target intracellular tau. And within neuropsych, I think one area that's not exactly new, but still extremely early, is using psychedelics for treatment of psychiatric diseases. And I remember, many, many years ago when I was a graduate student at Columbia, I remember sitting in the psychiatry grant rounds and seeing the ketamine data presented for the first time. And I remember, being very surprised first because the idea is pretty out there, but also, second, you can't really dispute the magnitude of effect there. And I think since then, the field has come a long way. And now we have a lot more data and we have products on the market. And both psychiatrists and the FDA has shown acceptance and even flexibility to both approving and using these drugs. And to my surprise, the FDA seems even more open-minded than some of the KOLs and psychiatrists. So I think we're still in the very early innings, but you're seeing a lot of new treatments in development, in very early stage development. And I think the key here is you want the right management team with the right background, which is traditional pharma drug development background, to bring these therapies to the market.
Super interesting. Thank you, Jingwen. Margarita?
Yeah, very hard to add to what's already been said. I think that covered the landscape. One thing is, I guess a couple of things we invested in, a couple of companies we invested in the past 12 months might be a little bit on the novel side is Nitrome, which is working on nitration and novel family of nitration enzymes, the lead program in Parkinson's, and also invested in Phase Medicines, which is investigating the role of molecular condensates in neurodegeneration. The other thing I think it could be really interesting is, looking at, in addition to I guess with relation to autophagy, the role of degraders and not necessarily protox, but maybe molecular glues or something. There's similar modalities.
Great, thank you. And Michael?
I think pretty much everything's been covered. I think Jingwen covered the psychedelic side, which I think is very fascinating. I think certainly you're seeing a lot of progress there, especially in TRD and other psychiatric disorders. I mean, in the last question, which I didn't get a chance to comment on, you think about the innovation here. 5 years ago, people wouldn't touch neuroscience. They wouldn't touch depression. They wouldn't touch really anything in the space. We've come a long way here. I think the momentum is great. And I think certainly you think about depression as one area where you have so many patients who are not served well and really are looking for a new treatment modality. To see really kind of the migration towards new novel mechanisms, certainly hitting the pathophysiology of the disease and now going to psychedelics, to me is very, very encouraging. So it's also good to see the strategic side of things, especially big pharma and big biotech make bets here. For a while, just given just the high placebo effect and just given this, you think about Phase 3 trial design, how hard it is, how expensive it is. People are looking more at cancer. Well, I think you're starting to see other areas, and it's cyclical. Look at vaccines, for example, with COVID. You see kind of the ups and downs or the vagaries of the marketplace. It's great to see people actually paying a lot more attention to this space.
Great, thank you. We do have a question from the audience. Maybe we could address this quickly if somebody wants to volunteer. It's a question about preclinical models and how investors gain confidence about translatability to the clinic and how you would evaluate a novel target that's only been tested in preclinical models. Would anyone like to volunteer to take that one?
I can speak to it. As someone who did her thesis using a few preclinical research models, I would say it's useful, but only to a certain extent. And there are also different preclinical models. So for example, preclinical models in psychiatry hold a lot less validity than some of the neurological preclinical models. And so that's why for me, unless the pathophysiology of the disease and how the drug target aligns really well, I rely heavily on clinical data.
Excellent, thank you for that. And I know we've gone into extra time here, but we do have one question remaining for the panelists. And that is, what advice would you give to early stage investors in neuroscience or entrepreneurs trying to raise capital for a new neuroscience venture? Michael, would you like to get us started?
Sure, look, I think being very focused is important here. I think especially as we migrate to more novel delivery systems, whether it's gene therapy and other areas, it's very costly. I think having enough cash in your balance sheet early on is important. Partnering with good people who are actually going to help you grow the company, I think it sounds trivial, but a lot of people make the mistake of really partnering with the wrong investors. I think that's really critical. I think getting to a go no go decision point, and that's my experience from big pharma, is really important. I think, the emotional aspect of drug development or drug discovery is really challenging. And you have to have a very objective look as you move forward. I think you also want to pick indications that are, I think represent a large medical need, and where pricing can be attractive, and also you can find a faster path to the marketplace. If you're a big company, you can afford to invest significantly in phase 2B and phase 3 trials. If you're a smaller company, even if you're well backed by strong VCs and crossovers and get public in an IPO, you still have a finite amount of capital. You got to be smart about how you use that. I mean, we're coming off probably the best biotech market we've all seen in our history. I mean, I've been doing this 25 years. I would say, kind of up until March, this was the best market I've seen as a banker, and also even before that. But I think times will change. I think you got to be really prudent and careful how you spend your money, because you think about companies going towards the public markets, there's still a finite amount of capital for drug development and for commercialization, especially.
Thank you, Michael. Margarita?
I think it's still about really differentiation, meaning, working on novel, really potentially transformational pathways in biology, not just, a single target, but looking at where your expertise is and focusing on that. Because I do think, to Michael's point, to be driving towards a phase 2 proof of concept is not something a biotech should be aiming at. What you want to do is be able to deliver really good data on a target that could be really exciting, differentiated from what's already on market and maybe even in the clinic. But I would echo those. It really is important to have the right investors. But biotech is, as we know, not linear. It's even less so with neuro. So need to find ones that are bold and not derailed by not afraid to make mistakes.
Great, thank you, Margarita. Jingwen?
Yeah, no, to me, it's really about what you're optimizing for and prioritizing for. So is it in terms of your portfolio prioritization or indication selection? So are you prioritizing because you really understand this disease and you understand what your drug target, or is it because you have very early clinical data that can point you to where to go in terms of indication selection? And that's not an easy task by any means.
Great, thank you, Jingwen. And Jamil?
Yeah, I would just add and maybe echo, neuroscience investing is not for the faint of heart. And so finding the right fund or syndicate to back your company is really critical. And seeing a track record of interest, expertise, and success in neuroscience, it takes a village to build a company. And so having really good groups around the table is critical. I think also, this is a long-term process. And so building relationships, building credibility with the investor, ecosystem, saying what you're going to do, doing it, bringing back that data. For us, at least as an early stage investor, it's not no, it's not yet. And these are the data sets that we want to see to gain that conviction, to take that early risk, to partner, to build a biotech company and hopefully develop transformational medicines for patients.
Excellent. We've gone slightly over time, so we'll end our panel discussion there. I want to thank our distinguished panelists again for graciously joining us today and sharing your insights on financing and investing in biotech companies that are focused on neuroscience. And with that, I'll turn it back over to you, Neil, to wrap things up.
Jay, thanks so much. That was really informative for entrepreneurs, and thank you for that. And I was channeling the advice just across my entrepreneurial aspirations. So thank you so much for that caboose on our train today. I put the salient information in the chat for those who see it. But for any other questions lingering from today, you can hit us up at clp@northwestern.edu. So that's the Chemistry of Life Processes Institute here at Northwestern, sponsors with BioCentury and Oppenheimer today. This concludes our program for today. I hope you take away lots of great facts, notes, networking contacts, etc. And it does sound like a village will be required to wrestle down these brain-based maladies. So a major challenge for us in our lifetimes. Let's rise to meet it. I wish you all a great week and indeed a great year. See you in 2022.